Welcome to today's event. My name is Simon Turner. I'm the publication manager for Cells, Tissues, Organs, and it's a great pleasure to welcome you to Karger's inaugural Young Investigator Award for the journal. Before handing over to Professor Ryan Gilbert, I would like to note that today's event will be recorded. I hope you'll enjoy today's event, and thank you very much for your participation. Professor Gilbert, I'll hand over to you now.
Okay, thank you, Simon. So hello, everyone. Welcome to the Young Investigator Award Symposium and webinar sponsored by the journal Cells, Tissues, Organs, which is a part of Karger publication. My name is Ryan Gilbert. I am one of the editors-in-chief of the journal. I'll be the facilitator for today's exciting event. Cells, Tissues, Organs, or also known as CTO, was founded by Karger publishers in 1945. Over the past 75 years, the journal has held extensive and rich history of publishing key manuscripts in diverse areas of investigation. The journal has changed with the implementation of a new editorial board, new editors-in-chief, and new publication managers at Karger Publishers. The journal's most recent focus is to bridge the gap between developmental biology and the emerging field of tissue engineering and regenerative medicine. Our most recent impact factor is 2.48, and that demonstrates a significant improvement over the past several years. And we currently have an acceptance rate of 33%, and our time to first decision for manuscripts completing peer review is 24 days. We welcome your submissions to our journal if they are in alignment with our areas of emphasis. In this next slide, I'd like to highlight our current editors-in-chief. For those of you considering CTO for your publication needs, we welcome your emails for further discussion on whether CTO is the right fit for your research. Again, my name is Ryan Gilbert. I'm the editor-in-chief of the Tissue Engineering Division of the journal, and I'm currently a professor and research scientist at Rensselaer Polytechnic Institute and the Albany Veterans Affairs Hospital, with over 20 years of experience in the field of tissue engineering. Dr. Gujun Sheng is another of our editors-in-chief. Dr. Sheng covers the stem cell biology aspect for CTO and is a professor at the International Research Center for Medical Sciences at Kumamoto University in Japan. Dr. Sheng's research focuses on epithelial to mesenchymal transition processes during mesoderm formation. And our third editor-in-chief is Dr. Christophe Vibon. Dr. Vibon is a professor of anatomy and embryology at the University of Göttingen in Germany. His research has focused on head and mesoderm induction during gastrulation and differentiation of primordial germ cells in the mammalian embryo. So he currently leads the developmental biology division of the journal. As you can see from the slide, we're responsible, the editors-in-chief are responsible for populating 5 to 6 issues per year, with manuscripts focusing on each of our focus areas, which is tissue engineering, stem cell biology, and developmental biology. And we also publish special topics issues. So in addition to the Young Investigator Awards issue, which we will highlight here today, we have published special topic issues led by our editorial board in diverse areas of interest, such as epithelial to mesenchymal transition, cell, tissue, and organ on a chip, human models of human disease, and bioartificial organs. We are looking forward to publishing several special topic issues in the future. So as always, we welcome your submissions to populate our issues per year, and we accept submissions that are diverse in format. We accept submissions that are research articles, review articles, systematic reviews, commentary articles, editorials, letter manuscripts, and methods articles. Format guidelines and specifications are located on the CTO website, which is posted here. You can also use the journal website as a link to our editorial board and editors-in-chief, and look up our most recent issues and ongoing special topic issues, including the Young Investigator Award competition. I'll now transition to one of our three editors-in-chief, Dr. Christophe Vibon, who will present some brief comments on the history of the journal and some additional perspective. Christophe.
Yes, hello. Thank you, Ryan, and hello to everybody. And first of all, congratulations to your nomination and awarding of the getting this award. And I hope you're not shocked by this picture of the front piece of the first issue in 1945, but I just wanted to take the opportunity to show my face and to let my voice be heard, so you know what's behind this, the name Christophe Vibon. And the reason I'm talking now is also because I found some interesting tradition in this journal, which for that I have got two quotes and one little piece of information, and I won't keep you long, but the quotes are very short, but I thought it's good to have. If you have a tradition, then it's good to know about it, because it may give inspiration to us, to all our colleagues who in their long haul enterprise of science, which has rare occasions of acknowledgement and success. You know all how difficult it is to conduct experimental work. So the first piece of quote is from the very first editorial, and it reads, of course, you know, it's all in the early days after the Second World War, and they take reference to that and say, science has suffered severely during a war of devastation. And then he goes on, there has been an ever-spreading increase in power of forces which have nothing to do with the objective search for truth. And I thought that was quite to the point for the last 4 or 5 years in the development of the world in various places. So we are still committed for search for the truth, or for new acknowledgement, new knowledge. And the second piece is further down this editorial, and it says, the scope of work covered by the journal includes the results of investigating tissues and cells by physical, chemical, genetical, and by developmental mechanical means. And that's, I think, is reflected by all your work that you're presenting today. You're using all these various methods, and you have a relevance to medicine and to morphology. And that's the third point, or a little piece of information. Anatomica, Acta Anatomica, you probably know, Anatomica comes from anatemnein, the Greek word for cutting. And that has the big advantage for our subject that there's no limit in the scope or topic that we cover, as long as it's somehow covered by structure or related to biological structures and can be made visible by figures, by pictures of such structures. So I think this Acta Anatomica has been always a focus and a possibility to publish varied results, and this since 75 years, and I'm very happy that you all of you continue to work in this line, and I hope this will indeed give you some inspiration. And with that, I thank you for the time you've given me for this. Thank you. Looking forward to your talks.
All right, thank you, Christophe. I'd also like to transition to Gujun Sheng, who also has some brief comments to share. Gujun?
Okay, can you guys hear me okay?
Yes.
Yeah, I don't have slides, but I think Ryan and Christophe have said it quite nicely. And CTO represents certain tradition of emphasis of a structural elements of things that we're all interested in, cells and tissues and organs, and how to build a human body, and how the disease state might be understood by anatomical analysis of cells and tissues and organs in a way. So I myself is interested in early development, and probably in the stage similar to what Christophe was mentioning. And I think we are very happy to try to build CTO as a kind of forward-looking and also engaging young researchers in a way. So we try to build a CTO as a community representing certain scientific ideology And I think we are very happy to have you participating and also possibly spreading the philosophy of CTO to your colleagues. And maybe we'll have more people joining this type of discussion. And with the overall aim is that science may be better served and by actually trying to figure out exactly what might be the essence that underlying the things that we are interested in, try to understand the biological mechanism for the normal state of either the animal development or homeostasis, and also the disease state that most of us are interested in anyway. So I think we will be very happy to have you present today, and also as a record. So we'll be posting the videos online, and actually they can take a look at what CTO might be interested in covering, and also your colleagues might understand what you are working with. CTO is trying to encourage submission from similar type of research outcomes in a way. So very much welcome, and also looking forward to actually listening to your presentations. Ryan, please.
Okay, thank you very much, Gujan. Okay, so let's move on to the reason why we're here today, and that's to highlight the work of awardees of our 2020-2021 Young Investigator Award competition. So again, the goal of the competition is to introduce new investigators to the journal, Cells, Tissues, Organs, and to highlight work of promising young investigators. Awardees were selected from a very competitive pool of applicants and were tasked with submitting a review manuscript or original research manuscript to a dedicated young investigator issue. So now we'll transition to our next slide. I want to briefly thank all the editors-in-chief for review of the applicants, and also to some of our associate editors, as you can see here, Loredana Di Bartolo, Casey Mara, Stefan Libau, for their contributions in helping us select the individuals for today's promising event. And also I'd like to thank Karger Publishers, which has really changed and become more forward-thinking, especially Dr. Simon Turner. Nadine has been great in terms of setting up the symposium, Denise, and also Angela. So let's move on to the awardees. So you can see the list of the awardees for the 2020-2021 year here. And I'd like to go through them each individually now. So from McGill University, I'm very pleased to present awardee Dr. Natasha Chang. Dr. Chang is an assistant professor in the Department of Biochemistry. And Dr. Chang, for the Young Investigator Award issue, she presented a review manuscript titled Empowering Muscle Stem Cells for the Treatment of Duchenne's Muscular Dystrophy. Congratulations to you, Dr. Chang. From the University of Miami, I'm very pleased to present awardee Dr. Courtney Dumont. Dr. Dumont is an assistant professor in the Department of Biomedical Engineering, and she contributed a research manuscript that's titled Biomaterial Strategies to Bolster Neural Stem Cell Mediated Repair of the Central Nervous System. Congratulations to you, Dr. Dumont. From the University of Pennsylvania, I'm very pleased to present awardee Dr. Ricardo Gottardi. Dr. Gottardi is an assistant professor in the Department of Bioengineering and the Department of Pediatrics, Division of Pulmonary Medicine. He contributed a research manuscript for the Young Investigator Award issue titled A High Throughput Mechanical Activator for Cartilage Engineering that enables rapid screening of in vitro response to tissue models to physiological and supraphysiological loads. So congratulations to Dr. Gottardi. From the Indian Institutes of Science, I'm very pleased to present Dr. Mohit Joly. Dr. Joly is an assistant professor at the Center for Biosystems Science and Engineering. Dr. Joly contributed an original research manuscript titled Computational Systems Biology Approach Identifies Slug as a Mediator for Partial Epithelial Mesenchymal Transition. So congratulations to Dr. Joly. Next, I'd like to congratulate from the University of California, Santa Barbara, Dr. Ryan Stowers. Dr. Stowers is an assistant professor in the Department of Biomedical and Mechanical Engineering, and he contributed a review article that's titled Advances in Extracellular Matrix Mimetic Hydrogels to Guide Stem Cell Fate. So congratulations to Dr. Stowers. From the University of Twente, I'm very pleased to present awardee Dr. Liliana Teixeira. She contributed a review article that's titled New Endeavors of Micro Tissue Engineering Cells, Tissue Organs on Chip and Communications thereof. So congratulations to Dr. Teixeira. And last, from the University of Freiburg, I'm very pleased to present awardee Dr. Peter Valentech. Dr. Valentech is the Emmy Noether Group Leader at the Department of Medicine at the University of Freiburg, and he contributed a review article titled Signaling Control of Mucociliary Epithelia, Stem Cell Fates, and Plasticity of Cell Identity in Development and Disease. So congratulations to Dr. Valentech. So you'll find our seminar schedule next. So please note that each awardee will receive 25 minutes for their talk, followed by a brief 5-minute question and answer session. So with that, I'd like to transition to our first awardee, Dr. Mohit Jolie.
Is my screen visible in full screen?
Yeah, looks great.
Thank you very much. I would like to begin with thanking all the editors-in-chief and the entire review committee of CTO for the Young Investigator Award. Thank you for the opportunity to present our work here at the symposium as well as the special issue. I think this is a fantastic initiative. So thank you once again. Today I'll be talking about the work that we published in the special issue, where we looked at mathematical modeling and integrated that with experimental data to try to understand the dynamics of this process of epithelial mesenchymal phenotype, particularly in the context of cancer metastasis. So Cancer Systems Biology Lab that I lead here at Indian Institute of Science in Bangalore, we specifically focus on developing such mechanism-based mathematical models in close collaboration with various experimental cancer biology groups as well as clinicians to try to have a better understanding of the dynamics of the process of cancer metastasis and how cancer cells develop resistance against drugs, because these two aspects still remain the major unsolved clinical challenges out there. So our goal is to have a predictive mathematical model which can be personalized as well, so that we can predict which therapy is going to more likely to be working for which set of patients based on a better understanding of how the dynamics of these processes is ongoing. So metastasis, as I mentioned, still remains the major cause of all cancer-related deaths and largely happens to this fantastic set of freeway that we have in our body, through which cancer cells from one organ can spread to multiple different organs. Now, thankfully, metastasis is actually a highly inefficient process with extremely high attrition rates. And if this were not the case, then you can imagine the havoc this particular process can cause in the context of different patients being afflicted by this disease. When we think of cancer, one of the first terms that comes to attention is the idea of mutations, which are genetic changes that are inherited throughout generations, and some of them are likely to increase the fitness of cancer cells to achieve different aspects. But when we think about metastasis, there has been so far no unique mutational signature that can be ascribed particularly to metastasis. Of course, there are some mutational signatures present in the primary tumor which are seen in metastasis as well, but during the process of metastasis itself, so far no unique new additional mutational signatures have yet been identified. And this makes a whole lot of sense if you think about the time scale of these processes. So metastasis, typically cells stay in circulation for a period of 1 to 2 weeks. And as you know, that the time scale of cell division is usually 24 hours. Now, if one has to think about how many divisions a cell will go through and the rate of error in DNA copying, and then you further have yet another bottleneck on whether a particular mutation would be helpful or not, then that is an extremely small possibility, and a given cell only has approximately 10 divisions for that to achieve. So clearly that does not necessarily help the cancer cells per se to gain any of the advantages that are needed during metastasis. The other aspect why mutations might not necessarily be the most efficient way of achieving this entire process is because mutations are irreversible in nature. Whereas when cancer cells have to adapt to the changing environment around them, that needs to happen in a reversible manner because they have to alter various of these aspects, be it adhesion, be it migration, be it the ability to evade the immune system or colonize another organ. So therefore, what I'm going to talk about today is in contrast to the commonly held genetic paradigm about cancer, which is heritable, irreversible, and deterministic in nature, a non-genetic paradigm particularly about the dynamics of cancer metastasis, which is a reversible process, stochastic, and not necessarily heritable as well. And this idea of non-genetic switching, non-phenotypic plasticity, which has now become a hallmark of metastasis, is something which is very fundamental to developmental biology, because here again, you have an individual cell which can give rise to multiple different phenotypes as it passes through this Paddington landscape of cell fates, as was again proposed right around the time the journal CTO started in 1940s. And we know today that cells can switch back and forth, specifically under certain external perturbations. And this is what is called as dedifferentiation or transdifferentiation. So this process where one genotype can lead to more than one phenotype, which is the central tenet of developmental biology, is becoming more common in the field of cancer as well, where we realize that in addition to genetic changes, there can be these reversible phenotypic changes that can be happening that can increase the fitness of cells during this entire process. So one of these aspects, which is again very well studied in developmental biology and has been studied extensively in cancer biology for the past 20 years, is the idea of epithelial to mesenchymal transition, or EMT. So the idea being that there are these epithelial cells we are talking about solid tumors here, and particularly carcinomas, which begin in epithelial organs. So these epithelial cells typically do not have the property to migrate or invade, and they're able to switch to a different phenotype, gain this property to migrate and invade, and that usually helps them to get out of the primary tumor. And once they have reached the other organ, they no longer need to migrate or invade, rather than need to actually settle down and proliferate and grow a new colony. And at that point of time, what is postulated is that they undergo an exact opposite transition called MET, mesenchymal to epithelial transition. And more recently, both EMT and MET simultaneously or put together have been referred to as epithelial mesenchymal plasticity in the literature. Now, when we started looking into this from a mathematical modeling perspective, first surveyed the literature and what was known about EMT in the field of, at least in cancer biology. And these are representative schematics taken from different review articles written over a decade. And what you see here is that EMT was usually seen as a very binary process. So cells are reported to be either epithelial or mesenchymal. There were some reports talking about metastable intermediate state, but the end point was an individual cell which was migrating and again sort of going back. So we started thinking more carefully about this and asked this question: is EMT really a binary process? And the kinds of experiments that existed, experimental data that existed, again in vitro in cancer biology, was that one would take epithelial cells and then treat it with a canonical EMT inducer, for instance, in the case of TGF-beta, and then come back after a week or 2 weeks and see that there are changes in morphology, there are changes in molecular markers, possibly functions, and say that EMT has happened. But as we very well know, if we have data at any 2 points and no idea about what happened in between, that is very limited information about how the entire process has evolved. And COVID pandemic waves across the world have been a fantastic example of that. So we wanted to ask, is EMT really a binary process? And then we started looking at what was known about what networks control EMT. And clearly, it's a highly complex process, and these networks can depend on the cell type you're looking at and various other factors. So how can mathematical modeling help? Again, I would like to draw an analogy to an example that we are quite familiar with, that of weather predictions. So today, we are at a stage where we can reasonably predict the weather at a given point of time, at a given point of space. And we actually trust these predictions to an extent that many of us make our travel plans according to this. But 100 years ago, it started as writing down some fundamental equations of weather forecast, which were based on ideal gas laws and Newton's second law of motion and so on and so forth. And over these century, what has happened is that the predictions made by these mathematical models have been compared against the experimental data, in this particular case, the weather data, collected at different time points, and the technology has improved there as well to collect it with higher precision and accuracy. And in this iterative process of going back and forth, improving the model and matching it with the experimental data, again, the weather data, we have gotten to this particular stage. So the goal is that can we get to a similar predictive understanding of the dynamics of metastasis as well. So mathematical models usually have the role of addressing several different types of questions. The ones I'm going to talk about today is what different dynamics are possible for a given network. If you know the different interactions, can you tell what are the different possible dynamical trajectories that cells can take? What if you overexpress or downexpress or knock down a particular factor? How does that change the dynamics? And eventually, can we control the dynamics? Can we, as engineers, can we construct systems such that we achieve a particular dynamic? In this case, reducing metastasis. So what we did, we started doing is we looked at EMT literature, what different molecules were reported across different cell lines and how were they connected with each other, and came up with this very simple model. Here, there are two transcription factors that induce a more mesenchymal phenotype, and these two microRNAs that induce a more epithelial phenotype. And the numbers that you see here represent the number of binding sites which was gained from experimental data. So these kinds of models represent some sort of quantitative input-output relationship. So when I say snail inhibits MIR34, so there is a decrease in the production rate of MIR34 when snail comes and binds to the promoter region of MIR34. So we can write down those canonical forms, and such kind of models have been extensively built for bacteria, yeast, and various other scenarios. We wanted to see if this approach can help better understand the cancer cell dynamics as well. So we wrote down a set of equations. I'll not bore you with the details, but the general fundamental concept of these equations is that every biological entity that you see here is being produced at some rate, is being degraded at some rate, and the rates of production and degradation are also being varied by these different interactions that you see here, which are represented. So I just converted this particular diagram into a more formal set of mathematical equations. Long story short, what did we find? So here the x-axis is some sort of an EMT inducing signal, and the y-axis is the measure of ZEB levels, which is the measure of EMT-ness. So what you can see here is there are three blue lines which represent stable states and two red lines which represent unstable states. So we only care about stable states because that's what will give us a phenotype. So we see an epithelial phenotype with very low levels of ZEB, a mesenchymal phenotype with very high levels of ZEB, but we also see a third state which has intermediate levels of ZEB as compared to epithelial and mesenchymal, saying that this is actually not a binary process. Now, if you look at the graph vertically upwards instead of horizontally, so there is a region of parameters here in which if you draw a vertical line upwards, you will cross only one solid blue line, which in this case corresponds to epithelial. Here, again, one solid blue line that corresponds to mesenchymal. But you do see regions in which you cross more than one solid blue line, which tells you that in this particular region, you can have cells in three different phenotypes, despite having identical genotype. So those were the three different predictions that we made, that A, EMT is not binary, B, if you have cells with the same genotype, you can still have three phenotypes, and three or more phenotypes, and the cells can actually switch back and forth between them, even without inducing them per se. So we collaborated with a set of lung cancer experimental cancer biologists, and here in this particular case, this is a cell line which has been stained for ecadherin and bimentin in green and red respectively. These are canonical epithelial and mesenchymal markers, And you see that individual cells do show both red and green markers. And this is not just a static image, because if you culture the cell line multiple times, you see this phenomena is being repeated, suggesting that this is a good model system for a stable hybrid phenotype. Then we quantified this further via FACS in cell lines coming from different cancers. And you do see that again, there is heterogeneity. There are cells which are E-cadherin positive only, dimension positive only, and double positive. These are the hybrid cells. And when you segregate these three subpopulations and just culture them independently, what you observe over a period of time, this was experiments done independently by another group, that they actually switch. So you're not giving them any EMT inducer, any MET inducer, but because of noise which exists in the system, cells can switch back and forth. So this mathematical modeling and various other analysis that has been done over the past decade has now shifted in cancer biology the idea of EMT from being a very binary kind of system to a system which has multiple different phenotypes in between. And importantly, the new hybrid phenotypes that have been obtained in vitro, identified in vitro, in vivo, and also in the clinic are actually the most aggressive ones. So we have been missing out on the most aggressive phenotypes by thinking of EMT mistakenly as a binary state earlier. And these are just examples of from the clinical data as well. So this is experimental in vivo mouse model data showing clearly that these cells which are somewhere in between are actually the ones which are most metastatic. And when you look at pathology images constructed in 3D by putting together various different 2D slices, we do see that single cell migration is actually extremely rare, if at all any, in cancer. And if you go back to the binary paradigm of EMT, there the two possibilities were don't migrate or migrate as single cells. But with this third state, the idea of collective migration also began to be looked at more carefully. So that now we ask the questions, what factors stabilize hybrid phenotype? So this is the same network that I showed you earlier, and there are various different assumptions in this network. So for instance, MIR34, it's a family of microRNAs. ZEB1 is a family of transcription factors, and so on. So at that point of time, 8-9 years ago, we lumped them all together. But now with more data coming out, we revisited that assumption and asked, is it really accurate to lump them all together, or do members of the same family can have different growths? So we particularly focused on looking at snail, SNE1 and SNE2, snail and slug, because there was more and more data coming out that these might be doing different things in the context of EMT. So again, in developmental biology context, there were reports about snail2 or slug, particularly associated with this partial EMT or a hybrid kind of phenotype, and more recent data in cancer as well, showing that snail2 is something that can specifically mark cells which are there in that hybrid phenotype. So we asked this question, is slug, first of all, snail2 or slug, can this be a marker for this hybrid phenotype? And can this just be a marker, or can it actually stabilize cells in that? So what we did is we took our network and now expanded our network to include slug. And snail and slug, interestingly, also, despite being members of the same family, inhibit each other. And this work was driven by two students in the lab, Shubha and Sarthak. So what you see here is this is the same diagram that I showed you earlier. Now that this diagram changes when you include slug. The major change is that now you have a region of parameters in which the only phenotype that is available is hybrid, which we did not have earlier. Earlier, hybrid was always coexisting with other epithelial and mesenchymal phenotypes. So suggesting that yes, slug can stabilize hybrid phenotypes. And then because this is a computer model, you can do all detailed kind of analysis. You can actually vary the strength of all these individual links and then see which link is actually playing a more important role. So here we identified that slug inhibiting MIR200 was playing an important role in stabilizing this hybrid state. So that was our specific prediction. Then we wanted to go back to experimental data and see if this prediction that slug is or snail2 is specifically stabilizing or maintaining cells in a hybrid phenotype, or is a weaker inducer of EMT as compared to snail was being seen in the data as well. So we looked at these are prostate cancer cells which have been induced to undergo EMT, and then the inducer has been taken back. So this is the data when cells are induced to undergo MET, the reversal of it. So they were induced to undergo EMT via snail1 as well as via snail2. And this is one transcriptomic based method to measure the extent of EMT. The more negative the value, the more mesenchymal it is. So clearly, as compared to control, when EMT is induced, you can see that the snail induces a weaker EMT as compared, snail 2 induces a weaker EMT as compared to snail 1. But when you withdraw it, they all go back to being epithelial. And we used other similar transcriptomic-based methods, and the result was the same in the same data set. So across different methods, we saw the same trend that slug is a weaker inducer of EMT. Then we looked at other cells. These were prostate cancer cells. We also looked at human mammary cells. Again, we see that the same method, the snail induces a stronger EMT than snail 2 as well as snail 3. And in human mammary cells also the trend was the same, that snail was stronger inducer as compared to other transcription factors. Around that time, there were also reports that because slug is a weaker inducer of EMT, you can have cells which are double positive, which maintain this epithelial marker and also have this mesenchymal marker present. So we wanted to see, we challenged our model. Can we explain this? That why do you see slug positive and E-cadherin positive cells, but not really snail positive and E-cadherin positive cells? So again, we included E-cadherin in our model and did these in silico experiments where we overexpress snail or you overexpress slug. So what you see is that when you overexpress them and when you overexpress snail, then the hybrid phenotype actually goes down. But when you overexpress slug, the hybrid phenotype actually increases. So clearly there is some difference in the roles that these different molecules are playing in the dynamics of EMT. We looked at association of levels of slug with overall survival in multiple different cancers, multiple different data sets, and do see this trend that the higher the levels of snail, the worse the survival, again indicating towards the more aggressive behavior of a hybrid phenotype rather than a completely mesenchymal phenotype. So one of the other questions we wanted to ask is that, is slug also contributing to drug resistance? So there were reports that snail and slug can mediate resistance to tamoxifen, which is the first targeted therapy that was developed, and it's given to ER-positive breast cancer patients because it goes and blocks estrogen receptor. So again, Sarthak asked these questions that first of all, there is this association reported between drivers of EMT and tamoxifen resistance, but do we understand this at a mechanistic process? Are these processes driving each other in bidirectional manner? And what about the behavior of hybrid cells? So again, we constructed a similar network to what I showed you earlier, simulated this over multiple different parameter regions, and these are actually heat maps taken from simulations. So what you see here is that if you sort of quantify this further based on EMT score, you clearly see 3 peaks. So there are epithelial cells, there are hybrid cells, and mesenchymal cells. And similarly, based on tamoxifen resistance scores, you can see the cells are either sensitive or resistant. When you put them together, you see that cells which are epithelial are predominantly sensitive. The cells which are mesenchymal is predominantly resistant. But hybrid cells can also be resistant. And so suggesting that, again, cells need not undergo a complete transition to gain this property of resistance. If they are hybrid, they have a good enough chance to resist these particular drugs. And this idea was supported by some clinical data analysis that we also recently looked at. The other predictions that our model was able to make is that if you overexpress Z, which means if you're driving EMT, you increase resistant population. And if you downexpress it, you decrease the resistant population. This is what we made. And again, we were able to see in multiple different cases that if you overexpress either breast cancer cells or other prostate cancer cells as well, in that case, we did see that there was a decrease in the estrogen response signaling pathway, suggesting that tamoxifen resistance is being acquired. We did another set of simulations that we overexpressed this particular molecule, which is responsible for sensitivity to tamoxifen. And we saw that when this particular molecule was increased, the epithelial population increased, and when this was silenced, the mesenchymal population increased. And this is what we saw yet again in experimental data, published experimental data, saying that when you silence ER alpha, the ESR1 gene, you see an increase in various different mesenchymal markers and also the signaling pathway itself, and a decrease in estrogen receptor. And the similar reports were seen when we looked at sub-cell lines derived either sensitive or resistant. So put together, what this suggests is that if you're going from E to M, you're driving sensitive to resistant. And if you're going from sensitive to resistant, you're driving E to M. So these processes are bidirectionally coupled with each other. And interestingly here, what we observed was that specifically when you focus on hybrid resistant cells and compare it to hybrid sensitive cells, because not all hybrid cells are resistant, there is almost an equal number of cells which are sort of split back between resistant and sensitive. Interestingly, the slug levels were higher in resistant but not in sensitive. So again, slug is not necessarily only marking the hybrid cells, but it might also help to identify the hybrid cells which are resistant to tamoxifen. And the experimental validation of this aspect is still ongoing in our collaborators' lab. So sort of putting together what we figure out through this integration of mathematical models and experimental data going back and forth multiple number of times is that EMT is not really a binary process. There can be cells in this hybrid state, and slug or snail 2 can play an important role in stabilizing them by inducing a weaker EMT, and they can also mark cells which are specifically drug resistant and can correlate with poor survival across cancers. Now, EMT, sort of some of the ongoing questions I want to highlight is that clearly EMT is a highly complex process because there are changes happening at morphological level, there are changes happening at molecular level, and there are changes happening at functional level. And we have not really reached a state where we can clearly say how these three different branches associate with each other, at least in the context of cancer metastasis. So we continue to address these kinds of questions through this approach, going back and forth between theory and experiment, is that how many hybrid states exist and what molecules stabilize those states, and what are the properties of these different states? Are all of them migratory? Are all of them therapy resistant? And which states are actually most relevant to target in different cancers? And eventually, can we control the system? Can we design optimal therapies by combining various things to target a population of EMT phenotypes, which are constantly switching back and forth because of noise in the system? So with that, I would like to thank the fantastic set of people I've had the fortune to work with, and these colleagues come from various different backgrounds, which is what brings out the beauty. They're oncologists, mathematicians, physicists, chemists, engineers, all speaking different languages, but sort of coming together to address this important question. And I would like to thank lab members, specifically Sarthak and Shubha, whose work I presented. And again, these lab members come from various different backgrounds, brainstorm and teach each other about either maths or biology and work together to get a better understanding of the entire process. Thank you so much for your attention. I'll be happy to take questions.
All right, thank you very much for that excellent talk, Dr. Jolie. Now we have a couple minutes where we can have a question and answer session. So any questions from the audience? Gujan?
Yeah, if nobody asks, I'm going to ask, but I can wait for other questions. May I ask?
Yeah, go ahead.
Okay, great. Yeah, so Mohit, very nice presentation. I think I have two questions. One's related to the kind of molecular mechanistic point of view, is that, you know, based on the structure or amino acid sequence or binding partners of a snail versus a slug, can you make sense out of what might be the difference that can explain the actual, you know, the EMT related difference between snail 1 and snail 2? And if someone were to do a kind of swapping kind of experiments to move parts of these snail 1 sequence into snail 2 and the other way around, is there anything like this done, and is there any understanding based on the molecular kind of, you know, analysis of the these two genes?
Yeah, thank you, Gujan. I think that's an excellent question. I'm not sure if people have swapped these regions, but what information exists is that snail is able to bind to the promoter region of E-cadherin more strongly as compared to that by slug. Now, what different, so it sort of narrows down that it has to, at least one part that is playing the role is its ability to bind to different targets, because snail and slug, again, belonging to the same family, have a large number of common targets. And the sequence is available, but I'm not sure if the detailed molecular studies has been done, but that is a direction that can be taken and we can get to a better understanding there.
So the other related question is that in the end, it's a regulator of cellular morphology, right? So you're using, for example, E-cadherin is one of the possible targets, and possibly other markers, cell biological markers that might be involved in in determining the E versus M or versus the hybrid state. So is it possible, I mean, since you are working on computational biology type of matters anyway, is it possible to use other types of descriptive terms purely based on, for example, cell biological parameters, rather than based on regulators of those cell biological parameters, to have a more precise kind of description of the what you consider as a possibly one or many of the hybrid states that might be existing, and then people can use that as a more unbiased way of looking at application-wise is cancer metastasis point of view, but cell biological point of view, people can also use these possible markers as a way of describing what kind of states these cells are in, right? I mean, I'm imagining that a combination of, let's say, different cadherings, doesn't have necessarily have to be cadhering, different types of intermediate filament and different types of matrix proteins, so these kind of things, or cell shape, cell size, volume, these type of other descriptors of the cell biology of either epithelial, mesenchymal, or hybrid state cells. Right?
So two of the markers that I know people are studying in more detail, including some of our collaborators, is cell shape, where they're trying to quantify cell shape using different aspects, be it the minor major axis ratio or ferrite diameter and so on. And the other is compressibility of cells. So those are two physical features which are being more quantitatively looked at. And what we are also doing is doing these image analysis based on these cell shaped from data that we have at different points of EMT induction for a given cell line. So that will help us identify how these features are also changing, both at a population level as well as at an individual cell level, when EMT is being induced. Now, how does that map onto the levels of gene regulatory network molecules is something that is that forms the next step of this analysis once we have a good pipeline for the morphological analysis in the first place. But that's an open question completely, that how does morphology function and gene expression signatures connect with each other.
Okay, yeah, thank you very much.
Thank you.
Okay, so maybe time for just one more question. Go ahead, Peter.
Yes, I was really wondering, so really beautiful data and interesting. Can you see, when you look at different types of tumor cells derived from different either epithelial or mesenchymal origins, do you see that they kind of like converge into this like hybrid state? And can you see trends beyond snail and slug expression levels that can direct you towards what were the triggers, or what are maybe intermediate steps, how different types of cells go into a kind of like common cancerous state? Very, very interesting work. Congratulations.
And thanks so much, Peter. So some of the work that has been done, where you start from either epithelial or mesenchymal, and then you see many of them comes to hybrid, is in the context of when you culture them in 3D. So apparently these hybrid cells have resistance to anoikis and are able to grow well in 3 dimensions and form tumor spheres. And that is one of the reasons that is proposed that why they are most metastatic, because they're able to survive in circulation, and also when they reach a distant organ, they are able to grow in suspension. Now, what are the cell shape features? One of the features is that perhaps they're able to maintain some cell-cell adhesion so that they receive some growth signals through each other. But is that necessary or sufficient is an open question. Thank you. Thank you.
Okay, great. Thank you again, Mohit. Wonderful talk. Okay, so let's transition to our next speaker. So let's have Liliana transition to her talk.
Hi, I would like to start by thanking you for this opportunity to present my work, and thank you very much for the award. I'm going to start sharing my screen. Just a moment. Okay, yeah, there we go. So still in the same line of the previous presenter, I'm going to give you a glimpse of other models that we can use for developing disease models, for developing drug screening methodologies. And the title of this presentation is The New Endeavors of Microtissue Engineering, and I'll focus on presenting you organs on chips. So first, I would like to just show you an overview of what organs on chips are, what do we use them for, and what are the current challenges. I will also give you an overview of my background, and then I'll focus on different showcases. So a little bit of work we have been doing on cancer on chip. Then I'll proceed with a little showcase on Longhorn Chip, and I'll finish with a joint on chip model. At the end, I would just like to give you also an overview on how we can make these platforms, give these platforms more of a high throughput vision, or how we can implement these more into a larger scale and make it more accessible for everyone. And then I'll finalize with some take-home messages. So just a little bit about myself. So my name is Liliana Moreira Teixeira. I'm an assistant professor at the University of Twente, and my background is in biology initially. So I worked on development of biomaterials for tissue engineering, mostly focused on cartilage repair as well as bone repair. So this was more of a macro tissue engineering stage, and later on I followed more into micro tissue engineering and entered the world of organs on chips. And I've been working on different models, as I mentioned before, so cancer, bone marrow, skin, lung, for example, and I'm going to show you just a few of these. So what are organs on chips? So as people see them, is the next generation of in vitro models due to their complexity. So as you can see here in this color scheme, the organs on chips are in purple. You can see that these platforms are able to give us an overview of or at least enable the native tissue architecture, so bring us closer to mimicking the native tissues. We can have different maturation stages, and we can use patient-derived cells. So this basically we can mimic much better the human conditions and have more predictive models, therefore. There's many physiological stimuli that can be included, from mechanical stimulation to electrical, and this is considered a very dynamic system. We can control perfusion of different fluids, for example, emulating blood circulation, and the culture times can be adjusted from short to long, so that's not a limitation, as well as putting different organs together, so different chips together, and therefore it will give you a perspective of a systemic-like interaction. This moment, some limitations are mostly related to the costs and the high throughput, but more and more people are investing in these platforms as a viable alternative to animal models, and therefore the costs start decreasing and the throughput starts increasing. So these models are engineered 3D tissues within microfluidic systems. So we can simulate the mechanics and the physiology of key organ functions and mimic the tissues and organs, also the communication of these, which are very important for different disease conditions. So why do we need these new models? Because typically the 2D systems are too simplistic, and the animal models, because animals are not fully representative of humans, of course, they do not give the answers we need to enable an effective clinical translation of treatments. So what can we do with these models? We can offer more pathophysiological relevant model instructive, create a more dynamic environment, very controllable because we use microfluidics. We can use them for drug development, screening for precision medicine, because different cell types can be used. So from different patients, and then take that information in and develop the treatments that are more tuned towards groups of patients, for example, and can be applied for a different domain. So not only for pharma, but also cosmetics and for environmental applications. The innovation that these systems are hoping to bring is that we will shift from symptom-based diagnostics to a more understanding the pathophysiological disease conditions, to understand that basis of the human conditions, and shift from non-translatable models, such as some of the animal models, towards more representative ones. And therefore, the treatments can be directed, can be more specific and more directive, and therefore increase the precision and effectiveness of what has been developed, and speed up this process towards clinics. So the microfluidic chips, these organs on chips, they incorporate, as I mentioned before, can incorporate different cell types. So it can enable a very complex co-culture system. We can implement different stimuli. It's typically connected to perfusion equipment, but not necessarily. And because it's normally a 3D configuration to mimic the native microenvironments, an ECM extracellular matrix can be incorporated and give this instructive information onto the cells and better represent the native organs or tissues. These platforms also enable an online monitoring, so we can have readouts at real time, and different designs are in place to enable specific readouts, as well as to enable the organization and spatial precision that each organ demands, and also the communication between the tissues and organs. And again, because it's microfluidics, we can control which stimulation we enable to these platforms, either mechanical, electrical, or chemical. We can create gradients, and we have a very intense control of the signals that we supply, as well as the ones secreted by the system. Many interactions can be emulated, so we can establish communication between the organs, establish communication with the immune system. So it's ideal to study very complex diseases, for example, cancer. And this is the first example that I want to talk about. So here we designed a chip that was, the goal was to assess the delivery of a typical drug for the treatment of colorectal cancer. So this drug was loaded in nanoparticles, and what we wanted to see was the diffusion of these nanoparticles into the core of the chip, which had cancer cells. So with this system, we could see the diffusion inwards and how this drug in different concentrations was affecting the cells. So basically leading to cell death. That was the goal of this design. And around the central core that has the cancer cells, we had a vascular-like channel to simulate our circulation. Therefore, we coated this channel with microvascular cells. So we see that also these microvascular cells also infiltrating into the tumor area, the tumor-like chamber. And this was a model that you can see as like a thin slice of a solid tumor, and it's ideal to test the diffusion of different drugs into it and show how it's affecting from the outside into the core. So ideal to test different concentrations and how it affects the human cells. So here you just see an overview how the diffusion would go, and you can see it's extremely fast. And this was just a model of a drug that we tested. Many others can be tested, or different drugs together, and it's ideal to test different concentrations, so it gives a high throughput perspective. Second model that I would like to show you is the Longhorn Chip. Actually, it's more the Alveoli on chip model. And here I want to give you an overview on how we can enable these platforms to be more instructive to the cells, to form more and more representative microenvironments. And this model was designed to mimic the physiology of the alveoli. So the cells organize in these structures differently, and therefore this kind of 3D configuration is essential for enabling the different cell types to differentiate. So I'll show you in a moment how we managed to get these cells to have a representative model in this configuration. And this model is compatible with various disease models, so we can recreate fibrosis as well as cancer, and have this alveoli on chip connected with different organs if necessary. What I'm going to show you does not have mechanical simulation, but then later on this was another study being done, and it can be adapted to have this input added to the system as well. So this was the work done by Danielle Batista at Maastricht University. And what she started doing was to look very deeply on how the morphological cues were given to the cells, and recapitulating this kind of morphology by having this semi-sphere instruction 3D configuration reproduced in a polycarbonate film, porous one, and then this film was incorporated into a chip. And here you just have an overview on how we can model the perfusion, how we can tune what the cells are feeling, the shear, for example, and adapt it to the best conditions possible, emulating as close as possible the native microenvironment. And in this study, Danielle used primary cells, and what we wanted to see was the differentiation of cells into alveolar type 1 cells as well as 2. As you can see here, we managed to achieve those 2 cell types, which were essential for the validation of the model in this case. But we also showed that this system enables the possibility of co-culture with microvascular cells. And here you see that we achieve a very uniform coating of both sides of this instructive membrane. So this gives us an insight on how we can get closer and closer to the native microenvironment just by providing this instructive physiology-inspired design. Next model I wanted to talk to you about is the joint on chip. So here is a sort of multi-tissue on chip system because the joint, as you can see here, it comprises different tissues. I'm going to talk about mainly cartilage, bone, and the synovial membrane, but there's more tissues involved, and some diseases are affected by communication of these tissues, so such as osteoarthritis, which affects over 300 million people worldwide, and this disease affects the movement of patients. It causes severe pain, and it's actually a disease that has no cure at this moment. So there's many treatments, but unfortunately, none of them effective enough to solve the problem. Mostly because we still do not know what causes this disease. We do not know the trigger, and therefore more representative models are essential for us to understand the communication between these tissues that comprise the joint, how these stimulations such as mechanical stress or inflammation, how these triggers affect the different tissues, and therefore start developing better treatments to reach the goal of having an effective cure. So basically, we wanted to put these tissues in communication and assess these triggers and check how basically the cells respond to these different stimuli. So first, I'm going to show you the cartilage on chip, and afterwards a little bit on the synovial membrane. So the idea is to put these two organs on chips in close communication, so establish a loop as well as linear, so we can see if we start with one system, how the other one is affected and the other way around. And here you just see an overview on what we looked at, the different cells involved. So the chondrocytes, for example, in the cartilage on chip, and the cells that are present on both tissues, for example, the macrophages, and see how this inflammatory response trigger the disease. So, for example, with the synovial membrane, how the fibroblasts start proliferating, for example, and the macrophages polarizing. So this is just how we designed the chips, what we had in mind, basically on the cartilage on chip platform. Here you see an actuation system with multi-chambers. So the idea was to reproduce the moving joints. So it's not a one push, it's basically a waveform movement that gives a different compressive instruction to the cells. And here what we wanted to see is how this mechanical loading was affecting the cells on the different areas. And what you can see here is basically a chip made of PDMS and these different chambers being actuated, how they affect the cells in the different areas of chip. So basically, these are some of the results obtained by PhD student Carlo. And initially, he assessed how these cells have, how the cells were affected by this situation in terms of viability. And what he saw was that with higher strains, with strains higher than 20%, for example, the viability started being compromised. And this is how, for example, we create these disease models to see how cells are affected, how these responses are being perceived by the different cells that we incorporate in the system. In this case was just chondrocytes, but you can already see here on the right side that we can analyze the cytokine release, and this you can see that in the difference between just a compressive operation of mechanical loading, it's different from a wave, which is more physiologically relevant. It already affects the release of these cytokines. And here you can see the example of TNF-alpha as well as IL-6 and how different they respond to the different compression loading schemes. Also, we could see this different effect on the compression in one mode or in a waveform. It also affects the matrix being deposited by the cell. So if we have a waveform, the response that we have is the higher production of collagen type 2, which is the major protein being produced by the chondrocytes in the cartilage. So shifting a little bit to the surrounding tissues of cartilage, this is an overview on the synovium on chip platform. And here again, we introduced a mechanical stimulation in a slightly different way. So we evaluated orbital shear stress, again, having a readout as the detection of pro-inflammatory cytokines. And that's how we created this disease model, just starting with, in this case, one cell type, synovial fibroblasts, to start simple, and then having these readouts also when we trigger the system with pro-inflammatory additions. So this work was done by the postdoc Susanna. And these platforms make sense if we want to establish a route that leads to utilization, broader utilization by pharma, so that people understand how the system works, how they can use it in a higher throughput, how we can implement sensors and readouts in the least invasive way possible. So here is just an example of an approach where we can increase the throughput dramatically, and the readouts by including sensors in modules, by electrical interfacing, and automatic liquid routing within these systems. So this ideally would be a system that is more and more standardized to make it more efficient as well as easier to use. So I just highlighted here some messages, some take-home messages that I think are quite important. So we have to have a balance between the complexity and the difficulty. So the instructive elements need to be there, but these platforms, at the end, they need to be understood and easy to use by different people, not just specialists. And the models are designed to fulfill a specific application. So we have a question, we make a design, and we need to understand also the model limitations. So as we heard many times, all models are wrong, but some are useful. So the idea is to have this key information on the chips, key instructive elements, and try to make these platforms as useful as possible, but understanding also their limitations. So the technology now is being directed to more and more acceptance by the regulatory entities and by industry, and therefore to have an easier translation and showing more and more validation, trying to show people that we can use these models to reduce animal use. Yes, it might be a bit too early to fully replace these animal studies, but we can definitely start reducing the numbers and refining the numbers of animals, and have a more representative platform that is easier to use by more and more people, more and more accepted from pharma to clinicians, for example. And with that, I would like to finish this presentation. I would like to thank all the students that contributed to these studies, as well as all the collaborators and the funding sources. If you're curious to hear more about organs on chips and what we have been developing at the University of Twente, please go to the website of the Organ on Chip Center Twente, have a look at the different models, how we have been using them. And also, if you are interested in pursuing an academic career in this area, there's open positions for a PhD student and a postdoctoral researcher. So with that, I would like to finish, and thank you once again for the award, and questions are welcome. Thank you so much.
All right, great. Thank you, Liliana. That was an excellent talk. I really appreciate it. So questions for the speaker? So Christophe?
I would have a question, but don't want to be in the way of anybody else. But as long as other people think about the question, what about the primary source of for your models? You once mentioned the primary cells for the alveoli. What do you use as a source for these models? Just the two cases, alveoli and perhaps the synovia. And would it be possible to take or to use induced stem cells?
Absolutely. So we started by using primary cells because we have access to the sources from different patients, also from the different disease models. So for example, for osteoarthritis, different grades of the disease. So we had that very well characterized, and we could validate the model better. But we can definitely establish the model using different cell sources, and iPSCs is one of the cell sources that is being more and more implemented in these systems, because you can have the genetic background also added, and it absolutely has added value. And a lot of systems are already in place and validated with these type of cells. So again, it's recreating the functional units, and the cell source is one of the parameters that you can easily define and have the different models using the different cell sources. Our choice.
Well, if I may ask a further question, what about the primary source for alveoli? How do you get hold of alveoli cells from the lung, from inside the lung? What is the source exactly?
So these ones were commercially available. So this was one of the studies developed by a consortium. The other partners were actually developing the iPSCs to have that input added to the system. So they have been characterizing these cells, and the next step was to replace the ones that we had been using, the primary ones, by the ones that have been developing and characterizing. So the publication will be out soon. And yeah, so I think that you have to balance also what you get, because then the maturation stages might vary. So that is the current stage of this project. Yeah.
Thank you.
Okay, great. Other questions? I have a question. I was very interested in your talk. So I work in the nervous system, and we're very interested in the blood-brain barrier. I was wondering if your group is working on this to have a better blood-brain barrier for central nervous system applications.
I do not do that personally, but there's quite a lot of groups working on that. And if you are curious about it, check the work that has been done by the different researchers of the center. So, for example, Andries van der Meer is focusing on that. Also, in the group of Alveoli van den Berg, they have quite a lot of models being developed, also incorporating different sensors. So these models are being developed at the University of Twente, and again, we develop models based on the research question we have. So we make our own platforms from design to validation, and I think that's key to have a more effective system to screen exactly what you want and better answer the question you propose to start with. So that's what we do at the university. Not me in this case. I mean, you cannot be the specialist in all organs, but we communicate and work closely together, because at the end, several diseases involve different organs. And we are in close contact to learn with each other and better implement this system also in a standardized way.
Great. And then maybe one final question for you. With these systems, I think it's really hard to have blood vasculature fully formed. And so when I usually see papers, I usually see blood vasculature just being endothelial cells. So the vessels haven't fully formed on the chip. Do you have any perspective as to how we might be better able to have vessels actually be amongst the cancer cells, as an example, so that we have a better representation of blood vasculature within these models?
So from what I learned so far and what I experienced so far was that the amount of cells that you put in to start with has a major role, as well as if you use microvasculature or cells or UVEX. I prefer always microvascular ones. The flow rates are also critical to have this coating as well. So you just have to understand how these cells attach, how long they need to start being stable, as well as at some points tune the medium so they don't start detaching. So that's also critical, that you understand the limit of the system. And for example, it's as easy as decreasing the amount of serum, for example, the tuning of the flow rate, having less shear, and the cells are more stably attached. So working also with different coatings. And I'm talking about, let's say, attachment to membranes. If you have a system where the infiltration of cells is necessary, the cells that attract these microvascular cells are fundamental. Again, you need to create these gradients so the cells have the instruction to go in. And that you can already have systems where you give just growth factors, even without the cells, to see how much would they need, and then start replacing by the amount of cells, the cancer cells, for example, and see at what point they start having this information, these cues, and start infiltrating inwards, and not having a vessel-like structure, but it's the infiltration into starts happening quite fast.
All right, great. Well, thank you again for a very excellent talk. So now we'll go to a brief break. So we have a break of around 10 minutes or so. So we'll see the schedule again here. So again, in around 10 minutes, you'll see that we'll start then with Peter's talk. So I'll see everyone back in around 10 minutes. Thank you. Okay, so let's, we have about a minute to go. So we'll start very soon. So far, excellent talks, great Young Investigator Symposium so far. So looking forward to the 3 remaining talks. All right, so why don't we begin? So I'll hand it over to Peter, who will give his talk now. Peter?
Now I can share my screen. 1 second. Here we go. I hope you can see me okay and hear me okay.
Yeah, looks great. Thank you.
All right. Wait, now it's gone. Here we go. So I would also like to thank the organizers and the selection committee for giving me the opportunity to present our work here. I'm just struggling here with the display settings. Give me 1 more second. I hope you can now see my presentation screen. So I would like to talk today about the signaling control of mucociliary epithelia, and specifically how we use frog tadpole. Yes,
Peter,
please.
we can't see your screen, your talk slides. I'm sorry.
Oh, that is unfortunate. Let me try 1 more time. Okay. Okay. Where is this? Can you see it now?
Yes.
Okay. All right.
Yes.
I hope.
Very good.
Yes. Okay. Sorry for this interruption. Okay. So I would like to talk about signaling control of mucociliary epithelia and how we use frog tadpoles to model airway diseases in humans. So we study mucociliary epithelia, which line various organs, most prominently the respiratory tract. And those mucociliary epithelia provide an important barrier against pathogens and infections. All mucociliary epithelia are constructed from basal tissue-specific stem cells, which reside on the base of the epithelium. They produce multiple secretory cell types, most importantly goblet mucus secretory cells that release mucus in the extracellular space. And they contain ciliated cells. So these are cells which have these hair-like structures projecting from their apical surface, and they beat in a coordinated fashion, which establishes extracellular fluid flows. So the protective function of this mucociliary epithelium, called airway clearance, works in such a way that when we breathe in particles and pathogens with our air, such as nasty viruses, they get trapped in the mucus layer, which is sticky, and then by means of mucus or by cilia beating, those are actually exported out of the respiratory tract to prevent infections and airway obstructions. So when this mucociliary transport breaks down, it increases the risk for patients for airway infections and obstructions, as I just said. And this is a feature in many chronic airway diseases. These diseases are thought to come from genetic predispositions, in addition to repetitive injury during lifetime, which changes behavior of epithelial and stem cells and leads to abnormal regeneration and differentiation of the epithelium, and this leads to either altered cell type composition, changes in cell function, or carcinogenic transformations of these tissues, and all of that leads to a breakdown of mucociliary clearance. And this is actually a serious global problem, because when we look at the WHO leading causes of death worldwide, actually 3 of the top 10 are related to airway epithelia, including chronic obstructive pulmonary disease as the number 3 cause of death worldwide. So in order to investigate which molecular mechanisms underlie these pathogenic changes and which potential therapeutic interventions we can develop to treat these diseases, we have different models to our disposal. Most importantly, we use human airway cells, either primary cells or immortalized human basal stem cells, and we can culture them in vitro. So some of the standard techniques used in the field are either planar L liquid interface cultures, where we can differentiate cells within about 2 to 3 weeks into an airway epithelium. More recently, we are also using 3D organoids to study mucociliary cell types and behavior. And in addition to that, we also take advantage of the power of mouse genetics to study mammalian lung function in vivo, as shown here in this example. In addition to that, with mice cells, we can do actually also all these approaches, extract cells and grow them in planar and 3D cultures. In addition to these well-established models, my lab actually focuses on an additional model, which is the frog Xenopus laevis, more precisely tadpoles of this frog, which generate a mucociliary epidermis. So they have an embryonic skin which resembles the mammalian airway in terms of cell type composition and function. But as you can see already here in these images, which are taken to scale, that these Xenopus cells are much, much bigger, and they are on the outside surface, so they are really easy to analyze and to manipulate. Furthermore, we have only 5 cell types, and the cell type composition is very stable, which allows us to study mechanisms, how this is established and maintained. We can generate very easily large quantities of mucociliary organoids to study cells, but also to harvest pure mucociliary tissues for systems biology approaches such as RNA-seq, attack-seq, chip-seq, or proteomics. And it is important to remember that actually key signaling mechanisms and pathways, as well as gene regulatory networks, are highly evolutionary conserved. So we can really learn from the Xenopus epidermis a lot about human physiology and pathophysiology. Since November is COPD Awareness Month, I would like to highlight 2 phenotypes and 2 studies that we conducted related to COPD. One phenotype is called Squamous Metaplegia. In this phenotype, we observe a loss of epithelial differentiation. Instead, we see hyperproliferation of basal cells that then stack and make multiple layers under the epithelium. And another frequently observed phenotype is called mucus or goblet cell hyperplasia. And in this phenotype, actually the balance between ciliated and secretory cells is tipped towards mucus secretory. And in both of these conditions, airway clearance does not function properly. So I would like to show you 2 vignettes of the lab, which were relatively recent published. One study driven by Maximilian Haas, a graduate student in my lab, relating to squamous metaplegia and Wnt signaling, as well as another study driven by Alexia Tasca, another grad student in my lab, which is dealing with Notch signaling and goblet cell hyperplasia. So let's start with the Wnt story first. So we study Wnt signaling in mucociliary epithelia for a while, and at some point we faced the conundrum that we looked at different studies, and different and conflicting roles for Wnt beta-catenin were proposed in mucociliary epithelia in different studies and in different models. So some said with Wnt downregulation we lose multiciliated cells, in others we get more multiciliated cells, and also the reverse condition, some upregulated Wnt signaling and get more ciliated cells. We didn't see any changes, and others reported that multiciliated cells were missing in these conditions. So we wondered, are these actually species and tissue-specific differences, or is there something else going on here? So we took a comparative approach first, and we studied Wnt signaling activation during mucociliary development. We initially started with the Xenopus mucociliary epidermis, which develops initially into two layers. The outer layer will give rise to mucus-secreting goblet cells, and the deep layer will become basal cells that then produce ciliated cells, ionocytes, and secretory cells. Those cells then intercalate into the outer epithelium. They differentiate cell type specific functions. And after 2 days, we have a fully differentiated mucociliary epithelium with mucociliary clearance. So taking advantage of a transgenic Wnt reporter signaling line in Xenopus, we actually analyzed Wnt signaling activity in these stages of mucociliary development. We saw that initially Wnt signaling was on in both layers and then got restricted to the basal cell layer. And during times of intercalation and maturation, we also saw individual multiciliated cells in the epithelium to be rich in Wnt signaling. And these signaling activation were maintained in mature epithelia. When we took the same approach using transgenic mice that have this Wnt reporter, and we analyzed similar stages in airway mucociliary development in this system, we could actually see that the patterns and signaling activities in both tissues and species were more or less the same. So this indicated that Wnt signaling is required in multiciliated cells and in basal stem cells, and that this was conserved across mucociliary epithelia and species. So next, we went on and we wanted to perform Wnt signaling loss of function. And in order to make it really Wnt beta-catenin specific, we used knockdown of beta-catenin directly. And we stained then for cilia here in green, and I hope that you can appreciate that loss of Wnt signaling and beta-catenin leads to an increased number of multiciliated cells in the epithelium, but these multiciliated cells actually fail in making normal cilia. Furthermore, we looked at the basal cell stem cell factor TP63, and we could see that simultaneously to this upregulation of multiciliated cells, we observed a loss of basal stem cells and the marker. And this indicated that really Wnt signaling is not only active, but actually regulates multiciliated and basal cells in mucociliary epithelia. Next, we took the opposite approach and we upregulated Wnt signaling using the small molecule bio. And I hope that you can appreciate that in here, this Wnt reporter frog line, we can really strongly overactivate Wnt signaling with this drug. And I hope you can also see that the basal cell layer actually starts to thickening. And this was indicative of basal cell hyperplasia like in COPD. So we confirmed this finding using our specific marker and this transcription factor for basal stem cells. And indeed, in these biotreated embryos, we saw a thickening of the basal cell layer. Simultaneously, when we looked at the mature epithelium, we could observe reduced or totally inhibited differentiation of epithelial cell types, thereby really recapitulating this basal cell hyperplasia and lack of differentiation phenotype. So we hypothesized that actually delta NTP63, which is a master transcription factor for basal cells, was a key regulator and mediator of this Wnt-induced inhibition of differentiation and hyperplasia. And therefore, we knocked down delta NTP63. And in these cases, we could actually rescue and prevent this inhibitory effect from overactivated Wnt signaling. So what we found in our study was that Wnt signaling activation acts at different levels of mucociliary cell types in ciliated and basal cells. And in basal cells, it activates delta NTP63, which then actually leads to overproliferation of basal cells and simultaneously blocks differentiation into epithelial cell types. So this also indicated that actually the observed conflicting roles for wind signaling in different models and studies were actually largely due to differences in the manipulation time point and tools rather than in differences of the biology and the signaling pathway functions. So since we found that this is the key mediator of this wind-induced inhibition, we were wondering if we can actually fix this situation. So to do that, we incubated our tadpoles and this bio-drug. We inhibited differentiation, we induced basal cell hyperplasia, and then we actually washed out the drug and gave the embryos a little bit of time to regenerate and checked these features again. And indeed, we observed that after just a couple of hours, differentiation of ciliated and secretory cell types was reestablished, and also the levels of TP63, the basal layer, got back to normal. So this was really promising also as a potential entry point into disease therapy, and therefore we next turned to immortalized human airway stem cells that we culture in air liquid interface conditions, and there we overactivated wind signaling using the agonist R-spondin-2. And indeed, we found that in this case, we also suppressed differentiation of ciliated and secretory cells, just like in Xenopus. And when we washed out this wind agonist and gave those cultures time to regenerate, we could actually trigger differentiation in human cells as well. And this was due, like in Xenopus, to the fact that we are overactivating basal stem cells. We checked this by TP63, but also by keratin 5 staining, a different marker for airway stem cells, which were both strongly upregulated in this wind-induced situation. But after regeneration and recovery, those actually got back to normal. And this indicated that wind beta-catenin signaling inhibition could be actually maybe used as a therapeutic intervention in some of these conditions. So to summarize this first part, what we could see was that there are multiple functions which are conserved in mucociliary epithelia of wind beta-catenin signaling. Wind signaling through activation of delta and TP63 drives basal cell hyperplasia, but this is reversible, and therefore blocking wind signalings and developing signaling inhibitors might actually be employed as a treatment in certain COPD and pre-malignant lesion conditions. So now let me switch to the other topic, the balance between ciliated and secretory cells and notch signaling. So we know that different notch levels are required for the different mucociliary cell types, in such that multiciliated cells are formed in low notch conditions, while secretory goblet cells are formed in notch high conditions. And this was actually first shown in Xenopus and later on validated in mammalian airways as well. We know from goblet cell hyperplasia or metaplegia that this is a combination between inflammation and increased notch signaling, and multiple studies have elucidated that it's actually a combinatorial inflammation input together with EGF signaling that drive JAK-STAT signaling. JAK-STAT signaling at high levels actually prevents apoptosis and also leads to a transformation of ciliated cells to mucous secretory cells, which is supported by notch signaling functions. But as in the wind study, there were some other studies that said that actually this transdifferentiation of ciliated to secretory cells in the mammalian airway is impossible, and therefore they questioned these mechanisms. So we took a look at our Xenopus epidermis, and we thought that we can maybe exploit the fact that it naturally remodels from a mucociliary epithelium.
Peter, you're muted.
I'm muted.
Now it's okay. Now it's good. Yeah, sure.
Okay. All right. So we wanted to exploit this natural remodeling from a mucociliary to a purely secretory epidermis during pre-metamorphosis and metamorphosis stages in the frog. And we could indeed observe that we lose successively over the time of development multiciliated cells from the epithelium while the other cell types were maintained. When we quantified the cell type proportions, we could actually see this significant loss of ciliated cells, significant increase in goblet cells, and no changes in the other cell types. And also in the frog system, this was driven by an increase in notch signaling, which was derived from the underlying mesoderm, in which notch ligand expression is transiently elevated during development. So indeed, we see that notch signaling increases and leads to a gain in goblet cells and loss of multiciliated cells, similar to the mammalian disease condition. Next, we looked in detail what happens to these cells, and we could actually find that while we found normal multiciliated cells, during these stages of change, we found these transdifferentiating multiciliated cells, which still possess cilia, but already started to produce and to secrete mucus granules. And their proportion really increased over the time of this transformation process. Furthermore, we inhibited notch signaling using the small molecule DAPT, and we could actually prevent this transformation from normal to transdifferentiating multiciliated cells. Furthermore, when we inhibited JAK-STAT signaling using the small molecule Ruxo, we could actually lead or switch from this transdifferentiation into apoptosis and shedding of the multiciliated cells, a process that was also proposed in the mammalian airway system. So we can conclude that actually the key regulation of this process by notch and JAK-STAT signaling is also conserved across species. So then we looked in detail what happens to these multiciliated cells during transdifferentiation, and we could actually observe that these cells retract and reduce the cilia, which was reminiscent of a process that we know from primary cilia retraction. Because when cells want to re-enter the cell cycle, they actually need to retract the cilium in order to repurpose the basal body as a centriole for during cytokinesis and chromosome separation. And these switches from ciliated to non-ciliated are driven by changes in proteostasis, a switch from autophagy to proteasomal degradation. So we wondered, could manipulations of the proteostasis prevent multiciliated cell and cilia loss in these conditions as well? And for that, we took advantage of two drugs. One of them is rapamycin. It inhibits mTOR signaling, which in turn inhibits autophagy. So by a rapamycin application, we increase autophagy. And by application of MG132, we actually block proteasomal degradation. So we applied those drugs to our tadpoles, and indeed in both of these conditions we could prevent the loss of multiciliated cells and cilia from the epithelium, indicating that during this transdifferentiation process these proteasomal and autophagy mechanisms are similarly purposed like during primary cilia retraction. And collectively, this indicates that rapamycin might actually pose a promising drug to counteract some COPD conditions in patients. So to summarize this part, we actually found two ways how the epithelium gets rid of multiciliated cells, through apoptosis or through transdifferentiation into secretory cells. This is induced by notch signaling, and we can prevent this by blocking either notch, JAK-STAT, or mTOR signaling. Furthermore, this also implicated that this goblet cell hyperplasia could be a reactivation of a normal developmental mechanism in the adult lung caused by inflammation, so reactivation of a gene regulatory network out of context, somewhat similar to cancerous conditions. If you now got interested in signaling and mucociliary epithelia as well as the diseases, I would like to encourage you to look into this review article where we summarize some of the recent findings and perspectives. And with this, I would like to thank our funding sources for giving us money to do this work. I would also specifically like to thank Maximilian Haas and Alexia Tasca, who were the main authors of the two studies presented, as well as Jose Luis and Magdalena for their help in these studies, to our collaborators on these two particular studies that I presented, as well as to additional collaborators on other projects. And with this, I thank you again very much for giving me the opportunity to present our work here, and I'm happy to take questions if there is enough time left. Thank you.
Great, thank you very much. Very interesting talk, Peter. Thank you. We do have time for some questions. Do we have questions from the audience?
May I ask a question?
Sure, go ahead, Gujan.
Very interesting, Peter. So I think the many aspects of that really look like the lung epithelial cells in the lung, I think, including the notch signaling, whatever. So a question I have is that how prominent is the phenomena of transdifferentiation from multiciliated cells to goblet cells? Is it the one way of is it main mechanism generating the goblet cells, or it's actually just some sort of response, special response for changing whatever dynamics of the skin? So
yeah, so in the frog, this is a normal way. So this is just something that happens during the normal process of tissue remodeling during metamorphosis. And there actually, it's probably just necessary to get rid of these multiciliated cells because the adult skin is a protective layer in amphibians, and this is strictly non-ciliated and just secretory. Furthermore, the tadpole and the adult frog uses a lateral line system to, orient itself in water currents. So if you would have an epithelium or an epidermis covered by cilia that make flow, I think this would make it very, very hard for the organism to orient itself in the water currents. And I think this is one of the reasons why they need to get rid of these ciliated cells eventually. In the mammalian condition, in the airway condition, actually transdifferentiation is surprisingly common because during homeostasis and regeneration, we know that, for example, multiciliated cells can be derived from club secretory cells. This is actually the normal mode during homeostasis, while these multiciliated cells during development are formed from basal stem cells. We also know from the literature that there is transdifferentiation of these club secretory cells into mucus goblet secretory cells in inflammation. And there are also reports, although conflicting, that a multiciliated cell can transdifferentiate into a secretory cell, but also a secretory cell, a goblet cell, into a ciliated cell. So it's really weird, actually, but also very, very interesting that while in many other organs, transdifferentiation processes are rather rarely observed, in the lung, in the homeostatic and regenerating lung, these processes are quite common and actually dominant, I would say, in certain conditions. And therefore, it's really, really interesting, but also important to look into the kind of like gene regulatory network, evolutionary speaking, where these abilities to change cell fates were coming from and how they are reactivated through inflammation or other processes in the adult organ, because in the adult organ, maybe the cells don't have the ability to respond in a similar purposeful and useful way as they do in tadpoles, right? And therefore, this can lead to disease because things are getting out of step and dysynchronized.
So how much of, so does it happen in a post-mitotic way? I mean, the cells do transdifferentiation after the terminal kind of mitosis, or they're still dividing cells in a way?
So certain cell types are known to be still able to divide, especially these club cells. They were actually thought for quite a while in the mammalian airway to be the stem cell population before actually the basal stem cells were described as bona fide stem cells of the epithelium. In general, these transdifferentiation processes are post-mitotic, so we have direct cell fate change from one terminally to another terminally differentiated cell type. Although I should say that in some very severe conditions where you delete a lot of surface epithelial cells, actually mesenchymal cells can be recruited and then differentiate into a new population or new generation of basal stem cells, which then can also regenerate the airway epithelium. So there is a lot of things going on, but for sure we observe a lot of post-mitotic terminally differentiated cells, which then are able to still switch their cell fate back and forth, actually.
Right. Ryan, can I ask one more question if there's nobody else?
We have a couple of questions, but go ahead if you have one more.
Yeah, well, very, very quick one. So, I mean, on your introductory slide, you mentioned something about EMT, I assume, right? Well, this is not in the context of the skin, of the tadpole skin, but I think there's some similar issues in terms of either remodeling, wound healing, and possibly, I don't know whether they form any kind of tumor or whatever. Is there an EMT issue involved? Could this be used as a model for studying EMT?
So I think that in certain conditions, it certainly can be used as an EMT process. In the Xenopus epidermis, we see it's maybe like an EMT-like process, but it's more similar to epithelial shedding. So we know that when we expose the cells either to high notch signaling without JAK-STAT input or other conditions, we can actually trigger this the apoptosis of the multiciliated cells, and they are usually shed from the epithelium. So they can either get kicked out of the epithelium, sometimes they are actually swallowed, but most of the time we think what we see is that they get eaten by macrophages or they just burst, and then we see other cells crawling around and eating the debris. So probably somebody like you studying real EMT would not call this real EMT, but maybe EMT-like processes.
Great. Yeah. Thank you very much.
You're welcome. Thanks.
Great. Thanks, Gujan. A couple of questions from the audience, Peter, that I'd like to share with you.
Yes, please.
You mentioned defective cilia formation after loss of Wnt beta-catenin function. What does it mean, is the question.
So this means that so we have a cascade of transcription factors which initiates cell fate in these multiciliated cells and then activates another layer of transcription factors which then drives ciliogenesis and cilia motility. So in the case of this beta-catenin loss of function approach, what we see is that the cell fate of multiciliated cells is still established. But because this other transcription factor, which is really necessary to make motile cilia, called FOXJ1, this one requires also Wnt signaling input to reach appropriate levels. So just the activation of the upstream transcription factors is not enough, but it needs some additional input from Wnt signaling. So in this condition, while we make an excess level of multiciliated cells, they still lack some of the Wnt input, which is required to drive FOXJ1 expression and really assemble these motile cilia. So we have more multiciliated cells, but in the end, probably fewer cilia that are functional to drive fluid flow.
Okay, great. And then one final, perhaps quick question. What parameters of ciliation did you analyze?
So in one study, we actually in the Wnt study, we just analyzed the quality of ciliation and the number of ciliated cells, but we looked in previous studies also on cell polarity, apical expansion, F-actin formation, and other features. In the NOTCH study, we looked in this transdifferentiation process for cilia acetylation, because deacetylation is a hallmark of cilia retraction. We also looked at the loss of specific basal body components. So we see that during this transdifferentiation process, some of the central components of basal bodies are still present, but for example, proteins decorating basal bodies that mediate the attachment to the apical membrane, like SEP164, they get lost. And by TEM studies, we actually could see a lot of clumps of basal bodies which look like they were kind of like half digested. So what we propose is that actually this proteostasis mechanisms, this degradation, actually targets specifically cilia and basal body proteins to get rid of them and make space for the secretory apparatus and to adopt the new cell fate, but also the new cell morphology required in goblet cells.
Great. Well, thanks again, Peter. Very interesting talk.
Thank you very much. My pleasure.
So now we'll switch over to Courtney Duma. So Courtney, take it away.
Can you see my screen?
Yes, we can. Thank you.
Perfect. Okay. So I'm Courtney Duma, an assistant professor of biomedical engineering here at the University of Miami. I wanted to start out just by thanking all the organizers for putting this symposium together, as well as the special edition of CTO. I especially want to thank the editors, Drs. Gilbert, Shang, and Vibon, for putting this together and giving both myself as well as all the other junior faculty an opportunity to showcase what we've been working on. So my lab is actually housed within biomedical engineering, but the physical space is within the Biomedical Nanotechnology Institute at the University of Miami, also referred to as Bionium. I won't be talking about any of the developments in nanotechnology that we've been working on today. Instead, I'm going to be showcasing the other half of my lab, which is housed within the Miami Project to Cure Paralysis here at the University of Miami. And along those lines, I'll be focusing on biomaterial strategies to bolster neurostem cell mediated repair of the injured spinal cord. So the spinal cord injury results, sorry, something just happened with my screen. So spinal cord injury results in loss of function to the nervous system below the level of injury. So if we're talking about high-level spinal cord injuries, everything below the level of injury, you'll see paralysis of. And typically, we're thinking of this as a quality of life condition in terms of your musculoskeletal system and sensory system losing functionality. But depending on the level of injury, you might also start to see loss of innervation to major organ systems. The most common dysfunction is loss of bladder function as well as sexual dysfunction. So these also factor in quality of life as well, but can also lead to life-threatening complications. So while spinal cord injury, if you look to the side here, is much lower relative to peripheral nerve injuries and traumatic brain injuries, they actually cause a disproportionate healthcare related cost. And this is due to the loss of mobility and the loss of independence of most of the people suffering from spinal cord injury, and why it's such a high priority area here in Miami at the Miami Project to Cure Paralysis. In order to develop strategies to address some of the challenges with spinal cord injury, we need to first look at what's happening at the tissue level. So here on the slide in green, we have axons extending uniaxially along the length of the spinal cord. And these are going to be conducting the signals and are ensheathed in myelin, which helps with conduction speed of signals along the axons. And so there's two main types of spinal cord injury. There's contusion injuries, which are you can think of as a compression injury that results in bruising. And while these are more prevalent, my lab for today will be focusing on penetrating spinal cord injuries, which is more of a cut injury. And this is because it's cleaner model to study new biomaterial strategies in. However, my lab's also focusing on contusion injuries and integrating what we've learned from penetrating spinal cord injury models into the contusion model as well. So in terms of penetrating spinal cord injuries, initially what you'll see is an immediate loss of axons and myelin. Over time, what you'll see happen is immune cells will migrate into the tissue, secrete cytokines that result in inflammation, which leads to further dieback of the axons and further loss of myelinating cells. In order to try to defend against this, the central nervous system, in particular astrocytes, will come in and try to wall off that inflammation through production of matrix, which is known as the glial scar. So combined, the inflammation and glial scar have a benefit initially, but their prolonged presence can actually impede regeneration of this tissue. So one strategy that we're focusing on is the development of biomaterials to address spinal cord injury. And they can be tuned to match the mechanical properties, have particular cell adhesion proteins. They can be tuned for degradation in order to accommodate the regeneration process. But primarily, we're interested in them because biomaterials tend to attenuate some of that scar formation and limit inflammation immediately after injury. They also provide an area for cell transplantation or therapeutic factor delivery. And this can be tuned through design of the biomaterial, inclusion of porosity, and other bulk properties of the material. And this is generally achieved through two classes of biomaterials. So the first class is our hydrogel biomaterials, where their viscoelastic properties are great because they can be tuned to match the mechanical properties of the spinal cord tissue, which is very, very soft. But typically, most hydrogels are an injectable that has a nanopore structure, which is not very amenable to rapid cell integration into that tissue unless it has a very rapid degradation rate. So one way to address this challenge is the formation of microgels. So here in purple, you can see the actual microgels that are formed. And then in this panel C, these microgels can be fused together through a secondary polymerization process to form a network which cells can readily move throughout, which is what's being shown in green. The next challenge with hydrogels is that there's not really any guidance cues in there. So because the spinal cord grows uniaxially, that's one of the main biomaterial factors that we like, or features that we like to try to include in order to expedite the recovery process. There are a number of groups, including Dr. Gilbert's group, that have looked into the inclusion of electrospun fibers into hydrogels and other cues to kind of start informing the recovery process, but they're not the level that some of the second class of biomaterials are at yet. So the second class are more these aligned structures, including aligned fibers, which can be used as bundles, or they can be rolled into these conduits, which will be transplanted into the injury site, typically a penetrating injury model, much like what we study in my lab, and can help guide the regeneration process. There's also preformed or molded bridges, where again, you see these channels which axons will grow down. And in this case in particular, there's a large amount of porosity to facilitate cell integration. So these are also a great option, but they're very limited in what you can apply them to. You need to know what the injury parameters are prior to transplantation. So what we came up with was the development of a hybrid hydrogel strategy, where we form these microgels, much like I showed a few slides earlier. They're approximately 50 microns in diameter. These microgels can then be annealed into bridge strategies, kind of like the ones we saw on the previous slide, or into individual tubes. The individual tubes are much smaller than the injury itself, so you can use as many or as few tubes as are needed and cut them to length, so you don't necessarily need to know the injury dimensions prior to the implantation surgery. These are what our general structures look like. So this is a cross-section of the tube, and you can see that it has an open lumen, which supports axons growing through the tubes. And it has this porous structure which allows for rapid cell integration into the tubes. And importantly, they're well within the range of CNS mechanical properties. Specifically, our microspheres are this lower bar here, and they have a much lower compressive modulus simply due to the fact that there's so much porosity, as well as a soft hydrogel material. So how do these tubes work? So again, we revisit our schematic of the spinal cord, and we have our uniaxial alignment. We could have an abnormal defect in which we would pack the tubes into the injury site, and they can be cut to length. And then the goal is to hopefully facilitate regrowth of axons, remyelination, and infiltration of other support cells. And so in this longitudinal cross section, we have axons in red and myelin in green. And in this center section of this injury site, we can see axons actually growing through the center of one of our tubes, showing that they actually are supporting this repair process. So these tubes are kind of the foundation of a number of other projects within our lab. So they can be implanted into the injury site and used to remediate the environment prior to stem cell transplantation. Stem cells can actually be integrated into the microgels themselves and used as an encapsulation technique. We can also deliver therapeutic factors from these biomaterials either locally or systemically using a complementary targeting system. In the interest of time today, I'm going to only focus on 2 of these. Specifically, I'm going to focus on kind of the base level of what we're trying to achieve with these gels in terms of their support of neural stem cell mediated repair, and that we can implant our biomaterials, allow for some remediation of the environment, and then do a revision surgery where we go in and transplant the stem cells. And this is in line with how stem cell transplants typically work, in which case you want to wait at least 1 to 2 weeks after injury before transplanting the stem cells in order to give them kind of the best shot for survival. Because if you transplant them immediately after injury, it results in pretty dramatic die-off right away. And then the second story I'll tell you about is combining this strategy with a therapeutic release. So as I mentioned, the first one I want to talk about is the biomaterial itself. And so this is predicated on some of the challenges that occur with stem cell administration to the injured spinal cord. So we want to deliver these stem cells because they can repopulate the injury site with the neurons and glial cells that are lost, but they also secrete a number of factors that can support regeneration. Unfortunately, they can't enact these duties unless they're able to survive. So typically we isolate our primary cells and expand them as free-floating neurospheres. We dissociate them into single cell suspension, and then they're either loaded onto material, or more commonly what you see is they're directly injected into the injury site. When they're injected into the injury site, you see immediate loss of cells just due to the shear forces that are exerted on the cells. Typically they're suspended in a saline, which does not have any sort of cell adhesion cues. So you see anoikis just due to the fact that there's nothing for these cells to bind onto. So already just due to this injection process, you have a pretty dramatic loss in your cell transplant population prior to it even reaching the tissue. Once it reaches the tissue, there's a lot of inflammation because it's an injury site. So you'll see further loss of your transplant at that point, and anything that's remaining is likely going to be lost to immune cell mediated repair mechanism, immune cell mediated removal mechanisms. And typically in the literature, we see between 1% and 4% survival about a week after transplantation. And we've been able to bump that quite a bit higher, and that's what I'll be telling you about today. And we've been able to achieve this through the use of biomaterials. So biomaterials can address pretty much all of these challenges. Specifically today, I'll be focusing on the challenges associated with cell adhesion and with the biochemical assault that arises after the cells reach the microenvironment. So here's the general overlay. Again, we have our injury, we implant our tubes. The goal is to allow for some degree of recovery and remediation, simply due to the fact that biomaterials can reduce inflammation and reduce glial scarring, at which point we go in, we transplant our stem cells 2 weeks after injury, and then 2 weeks later, we go in and recover the implant area and evaluate what's happening with our stem cells. And these are all done in mouse models, and our cells are from EGFP black 6 mice. So we have primary GFP cells that we can retrieve later on. And so when we look at them 2 weeks after transplantation, we have cells that were transplanted directly into the injury site, or cells that were transplanted directly into our biomaterial 2 weeks after injury. And we see that there's a significant increase in the number of cells that are surviving, both in terms of their cell density in the tissue, as well as the percent survival. So we're getting closer to 4% survival 2 weeks after transplantation. So the overall flow of this experiment was, again, we create the injury, we immediately implant our tube structures. We do BRDU pulses for 7 days, and we do a pulse each day and then allow for washout for 7 days. And this is essentially to target any proliferating cells in the tissue, including endogenous progenitor populations, so we can start to divide up what's happening with our exogenous cell transplants that we deliver on day 14, and then how that correlates to the endogenous cell populations. Again, after 2 weeks, we extract the tissue. So the first thing we wanted to look at in terms of our surviving cells was their progenitor phenotype. So nestin is a marker for immature spinal progenitor phenotypes. The EGFP are our transplanted cells, and BRDU are proliferating endogenous cells, and those that co-localize with nestin are our endogenous progenitor populations, whereas those that co-localize with EGFP and nestin are endogenous, exogenous spinal progenitor populations. And then we made sure that there was no overlap of our BRDU and EGFP, as there shouldn't be, since we allowed a week for BRDU washout prior to transplantation of our cell population. So when quantifying these, we do see a significant increase in nestin positive EGFP cells. So these are exogenous progenitor populations when transplanted directly into the tubes relative to directly into the tissue. However, we didn't see a significant difference between any of our endogenous progenitor populations, but they are there. So we weren't necessarily expecting a significant increase in endogenous progenitors. There are some mixed data out there demonstrating that cell transplants will recruit endogenous progenitors, but there's also papers out there that have said the exact opposite. So what we're seeing is kind of a bit all over the place in terms of the endogenous progenitor population. So we wanted to characterize these surviving cells further. Staining for the stem progenitor cells was on the previous slide. So we looked at neuroblasts, early neurons, and mature neurons. And we did this through staining of nestin in TIJ1. So nestin, again, will be a progenitor marker. TIJ1 is an early neuron marker, and cells that are positive for both are actually neuroblasts. So in B and E, we have our neuroblast populations. On the top, we have our exogenous cells, and on the bottom, we have the endogenous cells. For all of these phenotypes, we didn't see any significant difference in terms of the commitment to neural lineages of the endogenous cells, but for our exogenous cell populations, we don't see a difference with our neuroblasts, but when we look at nestin negative TIJ1 positive cell populations, which are early neurons, we do see a significant increase in terms of exogenous cells that were transplanted directly into the tubes rather than to the tissue itself. And this is carried over to our mature neuron populations, which are NEW-N positive, and which we didn't see any in the non-biomaterial conditions, either for exogenous or endogenous populations. But again, with exogenous cells, we saw a greater commitment to this neural lineage. So this is promising in that the biomaterial is supporting an increased commitment to the neural lineage, as well as an enhanced survival. So the next thing that we wanted to look at was if this is contributing to repair mechanisms, specifically regrowth of axons through the injury site and remyelination of those axons. So axons are in red, and these are transverse sections, and myelin is in green with a peripheral nervous system specific marker in blue, but I won't dig into that too deep here. Essentially, what we see is in all of these conditions, the only conditions that were significant were conditions that had tubes. So we're not seeing a significant increase in axon density or remyelination synergistically from the tubes and the stem cells. And this is likely due to the fact that there was already axons growing into the tubes at 2 weeks when we went back in and transplanted our stem cells. So a lot of what we're seeing is skewed towards the biomaterial being there for 4 weeks, whereas the stem cells are only there for 2 weeks. So long-term study or longer studies might help parse this out a bit further, but in terms of this early recovery, it's predominantly built around the tubes rather than the stem cells themselves. So just summarizing this story briefly, we did see an increase in survival, and the increase was similar to what others have reported 1 week after injury, and we're seeing it 2 weeks after injury, which is promising. Having the biomaterial present does contribute to neural cell lineages compared to just direct transplantation of stem cells that is more widely used across the field, and that the stem cells are not necessarily at this early stage contributing to axon growth or remyelination. The tubes and the biomaterial in general is playing more of a role here. So if we can use the biomaterial to promote repair, then the stem cells might have benefits that kind of factor in later on. Ultimately, though, we did discover through this study that the biomaterial platform is beneficial for neural stem cell delivery in this case, and that separating out the neural stem cell and the biomaterial delivery mechanisms actually has a benefit in this model. So the next thing we wanted to do was to see if we could further these benefits that we saw in the earlier study through therapeutic release from the biomaterial. So again, we have our injured spinal cord. We implant the tubes, this time with a therapeutic. Specifically, we're using lentivirus that leads to an overexpression of the cytokine IL-10, which is anti-inflammatory. We chose this because we have experience with it, but also because the lentivirus allows us to deliver it without modifying our biomaterial, whereas a localized delivery strategy of the protein IL-10 would have required additional kind of modifications of a system we already know works. So future studies would look at protein delivery rather than overexpression via lentivirus. But here, this was kind of a proof of concept that we could deliver IL-10 to further remediate inflammation with the biomaterial prior to stem cell transplantation. So again, at 2 weeks, we deliver our stem cells and evaluate survival, phenotype, and recovery mechanisms. So the first step was to make sure that our lentivirus was actually working at the injury site and was leading to consistent upregulation. So we performed in vivo imaging using the IVIS system and detecting bioluminescence. Specifically, we had an overexpression of FLuc, so luciferase, and we used that to characterize lentiviral activity over a 12-week period. And for at least 8 of the weeks, we saw a significant increase in lentiviral expression using the reporter gene. So we know that when we deliver our IL-10, we will have this increased expression profile. So we had, again, a very similar timeline as before. We add the biomaterial, which has lentivirus. We use the BRDU, but I won't get into the progenitor phenotypes just in the interest of time. And then at 14 days, we deliver our stem cells and characterize longer term studies in terms of regeneration and functional recovery. So here we have our green transplanted cells. This is our control injecting directly into the injury. This is directly into the biomaterial, and this is survival directly into the biomaterial with the IL-10 lentivirus 2 weeks after stem cell transplant, 4 weeks after injury. And again, we see another significant increase in our survival. And now we're up to almost 8% survival compared to less than 1% survival, which we were seeing by injecting the cells directly into the injury site 2 weeks after injury. So this is really promising. We seem to keep getting this doubling effect in terms of our cell survival, and increased cell survival should hopefully have therapeutic benefits. There we go. So again, we did look at all of the progenitor phenotypes, but I'm not going to get into that here. I just want to show you what we saw in terms of the histological repair. And so in white, we have NF200, which is the axon marker. We were seeing both clusters or bundles of axons in our tubes, as well as individual axons throughout. And when we quantified these, all of the conditions that received a therapeutic, whether it was tubes, stem cells, or IL-10 alone, were significantly improved compared to just the spinal cord injury condition. And then the triple delivery of stem cells and anti-inflammatory and tubes led to a significant increase even beyond those. What was really interesting is we're getting such robust axon elongation in our triple condition, whereas if we look at our spinal cord injury only condition, at 12 weeks post recovery, we're actually seeing levels that were achieved at 4 weeks when we did deliver therapeutics in our prior study. So this is a pretty substantial difference when you think about the recovery timelines. The last little piece that I want to share with you is the functional recovery component. So we performed ladder beam tests where we look at the rodent's ability to properly place their paw on rungs of the ladder as they move across it and quantify successful placements and misplacements. And the ability of the animals to successfully place their paw at 2 weeks is pretty minimal. Although even at this early time point, you start to see the triple conditions start to distinguish itself from the rest of the groups. But by 4 weeks, we see a significant increase in the triple condition compared to our spinal cord injury only. At 8 weeks, we see every condition that received a tube, the biomaterial, are significantly improved, and then by 12 weeks, all conditions are significant compared to the spinal cord injury. So what this tells us is that this combined strategy leads to a more rapid recovery, likely due to an increase in sparing, an increase in repair achieved through anti-inflammatories, biomaterials, and stem cells. So again, this divided approach of delivering the biomaterial separate from the stem cells can improve survival, and it can be exacerbated or increased rather with anti-inflammatories, and that this leads to enhanced recovery and tissue repair. So we're taking what we're learning here and we're developing new models to start building into the more widely accepted contusion injury model, as well as exploring some of the other areas that I highlighted earlier in my talk. So with that, I want to thank the students in my lab, particularly Andrew Sorello, who was instrumental in this work, as well as our collaborators both here at Miami and abroad, and our funding, particularly pilot funding that we received through the Frost Institute of Chemistry and Molecular Sciences. So with that, I'd be happy to take any questions. Apologize for going a little bit over time. Yeah.
Okay, great. Thanks, Courtney. So questions for Courtney. So I have a lot of questions, Courtney, but we don't have a lot of time. So I'll just start off maybe by asking two, if I can. So we're really interested in aligning materials in the spinal cord as well. And so my question is, how are you able to have your tubes or your microgels aligned in such a way to have such robust axonal regeneration?
Yeah, so our tubes are basically taking the idea of pre-molded bridges and just breaking it down to its individual units. So previously we were using 7-channel bridges, so now we can put 7 tubes in there. And essentially the lumen creates a void space that allows axons to grow down it. And it's not at the level of the neurites, obviously, although we have tried including electrospun fibers down the center as well, and there's a slight increase in recovery, but it wasn't significant in our hands. There were a variety of reasons why that could have been the case, but I think that giving the longitudinal cues is really expediting the rate at which recovery is happening. It's kind of tedious to use such a model, and we're actually based on feedback, moving more towards models that have less of that longitudinal alignment and more towards contusion injury models. But you know, I'd love to hear at some point how things are going in your lab, particularly anything that's following up on Dr. Rivet's work. Yeah.
Great. And then just one final question that I have. It seems like your stem cells are doing better with the presence of the material. The question that I had is, what are you doing to your gels to enable the stem cells to attach so that they're able to integrate better with that material?
Yeah, so we did initial pilot studies where we just looked at the difference between the inclusion of cell adhesion peptides like RGD and ICVAB, as well as no attachment proteins. And we did not see a difference, really. I think it's just because when we implant them, there's a lot of blood in the system, so a number of proteins already start to adhere onto the microspheres. So I'm assuming that's why we didn't see a difference. It would be great in the future to have more specific regions in the tubes that could foster, you know, down the lumen, we could have neuron attachment sites specifically like ICVAB, and then on the surrounding sites, maybe something that encourages specifically progenitors or myelinating cells. So that's something that we've considered as well.
Okay, great. Any other questions for Courtney? Okay, so if not, thank you again, Courtney. Excellent talk.
Thank you.
All right, so we'll go to our final speaker for today, and that will be Natasha.
Hi. Can you hear me okay? And can you see me?
Yes, we can hear you and see your slides, so you're all set.
Sorry, I couldn't find the buttons after I turned on my screen sharing. Okay, so hopefully I can advance. Yes, okay. So I wanted to thank Cells, Tissues, Organs, as well as Karger Publishers for giving me this opportunity to present my lab's work to you today. And for really giving us this platform for highlighting the work of young investigators in this field of regenerative biology and regenerative medicine. So as it was introduced, the model system that my lab uses is the skeletal muscle system to study aspects in stem cell biology. And the cells that we're interested in are these muscle resident stem cells called muscle stem cells, which are also known as satellite cells. And they are pictured here in this electron micrograph and also in this cartoon diagram. And what I find really interesting about this cell population is that they really represent a very rare population within the muscle tissue. So they represent around 5% of the total myonuclei. However, these are the cells that are really responsible for the efficient regenerative capacity of muscle tissue. And so these cells reside in a very specific niche. They're located between the basal lamina and the sarcolemma of the myofiber. And they are normally maintained in a state of quiescence during homeostasis, but they become activated in response to cues that are induced from exercise or injury, or even in the case of muscle diseases. And so then they activate and proliferate and generate the progenitors that are required to repair the muscle tissue. And so in my lab, we are interested in studying these regulatory mechanisms that are important for satellite cell biology, and more importantly, to understand how these mechanisms become impaired in the context of muscle diseases. And so this is the classical diagram showing how satellite cells undergo and commit through the myogenic differentiation program. And really, this is tightly regulated by a hierarchy of transcription factors. So satellite cells are marked by their expression of the paired box transcription factor PAX7. And as they commit to myogenesis, they begin to express myogenic regulatory factors such as MIF5, as well as MyoD. They then transition into this transiently amplifying cell population known as myoblasts. And upon sufficient generation of myoblasts, they then undergo terminal differentiation and begin to express late-stage differentiation markers such as myogenin as well as MER4. And then work from several labs over the last several years have demonstrated that the satellite cell population is actually a heterogeneous population and is made up of a minor population of satellite stem cells and a major population of actually already committed satellite progenitor cells. And so importantly, these two subpopulations have different differential gene expression. They express high levels of PAX7 and the cell surface marker CD34, but importantly, they have never before expressed MIF5. And as well, they also, these two cell populations have different functional capacities with respect to regeneration. And so importantly, as satellite stem cells, they are capable of undergoing self-renewal, and this can occur through two major mechanisms, through either symmetric or asymmetric cell division. And so pictured here, a MIF5 negative stem cell can undergo a planar symmetric cell division to generate two identical daughter stem cells. Alternatively, it can undergo asymmetric cell division along the apical basal axis, and this generates both a stem cell daughter as well as a committed daughter cell. And it's this progenitor daughter cell that, together with the bulk population of committed satellite progenitor cells, that then go on to expand and generate these progenitors that are necessary to facilitate the muscle repair. And so what we are beginning to appreciate in the field is that there's this delicate balance between symmetric and asymmetric cell divisions that's really critical for maintaining efficient muscle regeneration. And so I mentioned that asymmetric cell divisions are important for generating progenitors that then participate in muscle repair, but equally important are these symmetric cell divisions because they are the ones that will maintain the stem cell pool. And this is really critical for sustaining the regenerative capacity throughout life. And what we now appreciate is that impairments or defects in this balance is an underlying cause for muscle degeneration. And so, for example, in aging, what we see is this increased commitment. So we see an increase towards asymmetric cell divisions and a loss of these symmetric stem cell divisions. And this ultimately leads to stem cell decline and stem cell depletion. And over time, this results in reduced progenitor generation. And interestingly, we see the exact opposite in the context of a muscle disease known as Duchenne muscular dystrophy. And this is what the focus of my talk will be on. And so we actually see in Duchenne muscular dystrophy a loss of these asymmetric cell divisions, leading to reduced progenitor generation. And interestingly, an increase in symmetric cell divisions, which leads to stem cell hyperplasia. However, these stem cells are dysfunctional. And I'll go on and talk a little bit more about the molecular mechanisms of this stem cell dysfunction. But first, as I mentioned, this is the disease that my talk will focus on, and it's also a topic of research focus in my lab. So this is Duchenne muscular dystrophy, which is also known as DMD. It's a lethal genetic disease that affects around 1 in every 3,500 boys worldwide, and it's characterized by progressive muscle wasting. which leads to severe loss of mobility and ambulation, and also loss of life around the ages of 20 to 30. And this is usually due to respiratory and cardiac complications. And importantly, there is no cure for DMD. And it's a genetic disease caused by mutations in the DMD gene, which encode for the protein dystrophin. So dystrophin is this protein here, and it's part of a large protein complex known as the dystrophin-associated glycoprotein complex, or the DGC. And the DGC is normally located at the myofiber membrane. What happens in the absence of dystrophin, which is what happens in DMD, this complex does not get assembled properly, and this leads to weakening of the myofiber membrane and increased susceptibility to contraction-induced damage. And so it has been widely accepted for a long time that this was the primary cause for DMD, this loss of myofiber membrane integrity, leading to muscle degeneration. However, recent work in our field have now highlighted a role for dystrophin in satellite cells. And this implicated satellite cells in DMD disease progression, suggesting that DMD is also a muscle stem cell disease. And so this is the work from Michael Rudnicki's group, and this was published in 2015. And it was shown that dystrophin is actually expressed in satellite cells, and it plays a very important role in wild-type satellite cells in mediating the establishment of cell polarity. And establishing cell polarity is a prerequisite for asymmetric cell division. And in the context of DMD, so they used a mouse model, the MDX mouse, which is a mouse model for DMD that lacks dystrophin expression. What they found was that these satellite cells were unable to establish cell polarity, and this led to an inability of these cells to undergo asymmetric cell division. And as I mentioned, this results in the reduced generation of myogenic progenitors, which then contribute to this regeneration deficit in dystrophic muscle. And as I mentioned, we do see that these cells are able to undergo symmetric cell division, and we actually see enhanced numbers of stem cells in dystrophic muscle, both in human and in mouse DMD models. However, these stem cells are not functioning properly because they can't undergo asymmetric cell division and they can't generate the progenitors. And so this is the working model for how my lab views DMD disease progression, and that we envision it as a two-pronged disease. So lack of dystrophin protein is affecting the myofiber. It causes myofiber instability and increases myofiber damage. However, there's also this aspect of the disease where lack of dystrophin in satellite cells is causing satellite cell dysfunction, and this really exacerbates the disease by contributing to the inefficient muscle repair, but also it can contribute dysfunctional components from the satellite cell to the myofiber. And so we see both of these as contributing components to DMD disease progression. And this is really the focus of the review article that we submitted to Cells, Tissue, Organs, is to really highlight this evidence for a satellite cell impairment in DMD. And so I already mentioned the impaired asymmetric cell division from the Rudnicki lab, but previous work had also demonstrated using both human and mouse myogenic progenitors from DMD patients and DMD models that these progenitors exhibit altered myogenic kinetics. There's also a recent paper in bioRxiv showing that dystrophin deficient satellite cells exhibit dysfunctional mitochondria, and they can actually contribute these defective mitochondria to the myofiber upon fusion to the myofiber. Also, dystrophin deficient satellite cells exhibit alterations in the metabolic profile, and they also exhibit enhanced entry into cellular senescence, which is a state of irreversible cell cycle arrest. And so all this evidence really points to a role for satellite cells in DMD. And today I'm going to focus on one story where we were really, it really started out as a fundamental question, looking at how satellite cells regulate asymmetric cell division. And this provided insights into how this mechanism becomes impaired in the context of DMD. And so as I mentioned, we started out just looking for how do satellite cells regulate asymmetric satellite cell division, and how does this occur downstream of the dystrophin complex. And so what we were really interested in understanding is how does the satellite cell know to turn on MIF5 transcription in the progenitor cell, and how does it know to keep it off in the satellite stem cell? And so what's regulating this, especially following an asymmetric cell division? And so to look at this, we focused on this protein here, which is an arginine methyltransferase called CARM1. And the reason why we focused on this is because we knew that it was an important mediator of asymmetric cell division. And this is really building on previous work from Michael Rudnicki's group, showing that following an asymmetric satellite stem cell division, that this arginine methyltransferase CARM1 can directly methylate PAX7. And that methylation of PAX7 is required for PAX7 then to be able to bind and recruit this histone methyltransferase complex to the proximal promoter of MIF5. And this results in H3K4 trimethylation and opening of the chromatin and activation of MIF5 transcription. And this is occurring in the progenitor cell following an asymmetric cell division. And then so we asked the question, how is CARM1 being regulated during this process? And so we first started by performing a kinase screen to look for kinases that could phosphorylate CARM1 to identify potential regulators. And from our kinase screen, we pulled out P38 gamma MAP kinase as a candidate regulatory kinase. And so what I'm showing you here is an in vitro kinase assay where we incubated affinity purified P38 gamma kinase together with recombinant GST CARM1 protein. And what you can see is that in the presence of activated P38 gamma kinase, we see a very specific phosphorylation of CARM1 protein. So then we asked, do P38 gamma and CARM1 proteins, are they interacting actually in satellite cells? And so to address this, we performed proximity ligation analysis, which is an antibody-based approach that allows you to identify protein-protein interactions by immunofluorescence. And so here, the PLA signal is in red, and our satellite cells, which are cultured here on single EDL myofibers, are marked by expression of PAX7 in green. And what you can see is that in quiescent satellite cells, we did not see much of a PLA signal. However, we saw a very intense PLA signal at after 42 hours of culturing. So these are activated satellite cells. And what's nice about PLA is that you can then visualize where these protein interactions are occurring in the cell. And we see this very nice interaction signal at the cell periphery of activated satellite cells. So we next performed mass spectrometry to identify the exact site on CARM1 that's phosphorylated by P38 gamma. And we identified a single phosphopeptide that revealed a phosphorylated serine residue at the extreme C-terminus of CARM1. And so this is serine residue 572, and it is a novel phosphorylation site on CARM1. And importantly, it's also consistent with a P38 phosphorylation motif. So that's a phosphorylated serine or a threonine that's immediately followed by a proline. And when we compare various CARM1 sequences from different species, we saw that the serine was highly conserved. So next, we wanted to confirm that this was the site on CARM1 that's phosphorylated by P38 gamma. So we performed site-directed mutagenesis to generate a serine to alanine mutation at the site. And then we purified CARM1 protein that harbored this mutation. And then we performed in vitro kinase assays with P38 gamma. And what you can see is that P38 gamma can phosphorylate the wild-type CARM1 protein, but this phosphorylation is completely lost in our S572A mutant. And so this confirmed that this was the site that's phosphorylated by P38 gamma. And then we turn to PLA again to ask whether we can detect CARM1 phosphorylation at this site in satellite cells. And so here we made use of an antibody that recognizes this P38 phosphorylation motif together with a CARM1 antibody. And when we perform this PLA reaction in wild-type satellite cells, we see this very nice PLA signal suggesting that CARM1 in satellite cells is phosphorylated at this motif. And then we wanted to know if this phosphorylation was dependent on P38 gamma. So what we did was we used a genetic approach and deleted P38 gamma in satellite cells and then performed the same PLA in the absence of P38 gamma expression, and we saw a significant reduction in the PLA. So this suggested to us that phosphorylation of CARM1 at this motif is dependent on expression of P38 gamma kinase. So then we wanted to know what is the functional consequence of this phosphorylation event. And so again, we used site-directed mutagenesis, and we generated the serine to alanine mutant, which inhibits the phosphorylation on CARM1. We also generated a serine to glutamic acid mutant that will then mimic the phosphorylation on CARM1. And then we transfected these mutants together with wild-type CARM1 into CARM1 knockout primary myoblasts, and then we performed immunofluorescence to look at CARM1 subcellular localization. So CARM1 is normally localized both to the cytoplasm and to the nucleus, and that's what we see with our wild-type CARM1. And we also observe the same expression pattern with our S572A mutant. But what was really interesting and what we didn't expect to see was that in our serine to glutamic acid mutant, that we saw many of our cells exhibiting this predominantly cytoplasmic CARM1 distribution. And so this suggested to us that phosphorylation of CARM1 at this site was restricting CARM1 localization to the cytoplasm and preventing its nuclear translocation. And so this is our working model for the data that I've showed you thus far, that we knew that P38 gamma interacts with CARM1, and this is happening at the cell periphery, and that phosphorylation of CARM1 is restricting CARM1 localization in the cytoplasm and preventing its nuclear translocation, where it would normally methylate PAX7 and play a role in activating expression of myogenic commitment genes. So we asked then, how is P38 gamma regulated during this process? And we hypothesized that perhaps P38 gamma could be regulating with the dystrophin protein complex. And the rationale for this hypothesis was based on this publication from 1999, demonstrating that P38 gamma kinase can actually interact with the dystrophin complex through a direct protein interaction with alpha-1 syntrophin. And so we asked whether this interaction was also occurring in satellite cells, and based on RNA sequencing data that we had, we knew that the main isoforms of syntrophin that was expressed in satellite cells is beta-1 syntrophin. And so we asked whether P38 gamma could be interacting directly with the DGC through an interaction with beta-1 syntrophin. And so we again used proximity ligation analysis, this time with beta-1 syntrophin and P38 gamma antibodies, and we were able to detect a very nice PLA signal in activated satellite cells. And we often found that this interaction signal was polarized in these early activated satellite cells. So this suggested that P38 gamma does interact with beta-1 syntrophin in satellite cells. And then we asked, how is this affected in the context of DMD? So in the absence of dystrophin expression, the DGC does not get assembled. We asked what happens to this interaction. And so we performed the same PLA analysis, this time in satellite cells from MDX mice. Again, this is our mouse model for DMD, which don't express dystrophin. And what we found was that this interaction between beta-1 syntrophin and p38 gamma was completely lost in dystrophin deficient satellite cells. And this led us to hypothesize that the activity of p38 gamma is dysregulated in the absence of dystrophin. And to test this hypothesis, we then looked at the phosphorylation status of CARM1 in our MDX satellite cells. So we performed our same phospho-CARM1 PLA comparing wild type and MDX satellite cells, and we saw an enhanced PLA signal in our MDX satellite cells, suggesting that in the absence of dystrophin, we see hyperphosphorylation of CARM1. And we were able to quantify this by measuring fluorescence intensity, and it was significantly different in significantly higher in MDX satellite cells compared to wild type satellite cells. And so hyperphosphorylation of CARM1 would suggest that CARM1 nuclear translocation is impaired, which would then result in reduced methylation of PAX7, and this would then affect the ability of PAX7 to recruit the histone-methyltransferase complex that would then result in H3K4 trimethylation at MIF5. And so to directly test this, we then performed chromatin immunoprecipitation for the H3K4 trimethylation mark at MIF5 in primary myoblasts isolated from wild type, here shown in red, and in MDX cells shown in blue. And what we found was that in MDX cells, we saw significantly lower levels of H3K4 trimethylation at MIF5. And so then this would suggest that transcriptional activation of MIF5 is impaired in DMD cells. And so we tested this by performing RT-qPCR to look at levels of MIF5 transcript. And we did this both in freshly sorted satellite cells as well as cultured primary myoblasts derived from both wild type and MDX mice. And what we saw using both approaches is we saw reduced levels of MIF5 transcript in MDX cells compared to wild type cells. And then finally, we asked whether we can then rescue this impairment in MIF5 transcription if we inhibit p38 gamma. And so to do this, we use an siRNA approach. We isolated satellite cells from wild type and MDX mice, and then we treated them with p38 gamma siRNA. We did 2 rounds of transfection and then collected the cells to look at levels of transcript. And so you can see that we get very efficient knockdown of p38 gamma, both in wild type and in MDX cells. And what we were really happy to see is that we can see an increase in MIF5 transcript levels when we inhibit p38 gamma, both in wild type and MDX, but more importantly in the MDX cells where we saw lower levels of MIF5 transcript to begin with, we see a more pronounced increase in MIF5 levels, specifically in the MDX cells. And so to summarize, what I hope to have conveyed to you today is that through studying a very fundamental mechanism, we basically identified a signaling pathway that involves the dystrophin protein complex and its ability to regulate this signaling axis that involves p38 gamma and CARM1 to regulate the ability of CARM1 to facilitate asymmetric cell division. And importantly, this led us to, this provided insight into what's happening in the context of dystrophin deficiency. And so we were able to identify that in the absence of a DGC, that p38 gamma activity is impaired, and this leads to hyperphosphorylation of CARM1. And this results in reduced CARM1 translocation and reduced expression of myogenic commitment genes. So this provided a molecular explanation for the stem cell dysfunction that we observe in DMD, and also provides new opportunities for investigating potential avenues for therapy to treat DMD and restore this stem cell dysfunction. And so just to bring you back to this slide, that's really the focus of the research in my lab at McGill University. We are interested in understanding the fundamental mechanisms regulating satellite cell biology to understand how these mechanisms are impaired. in disease. And then finally, to understand how we can then use this information to then restore and correct this stem cell dysfunction to improve endogenous muscle repair for the treatment of muscle diseases. So I'll stop there, and I'd like to thank members of the Microfluidic lab for their contribution to the CARM1 story. This is my lab at McGill University. These are my graduates and undergraduate students. I want to thank also our research collaborators for contributing to the various research efforts in my lab, to our funding sources, and thank you for your attention. Don't hesitate to email me and reach out if you have additional questions, and obviously you can follow my lab's work through our website and through Twitter. So thank you very much.
Great. Thank you very much, Natasha. Questions for Natasha? Always the end of the session, maybe some questions will come in. Okay, Christophe, go ahead.
Yeah, Natasha, very elegant work, very nice, and not being in that subject at all, but I'm wondering whether this pathway is used in any other tissue in the body, or is it just really muscle?
So that's a good question, and it's not something that we've looked at, but I know that dystrophin, I believe dystrophin is also expressed in the brain, and there it is responsible for some of the neurological defects that have been observed in DMD patients. But we haven't looked at neural stem cells, for example, to see whether this mechanism is also playing a role in neural stem cells. But what I think is maybe amenable to other stem cell systems is this idea that there is a protein complex that's involved in regulating signaling pathways important for transcriptional activation of commitment genes, for example.
Yeah, thanks.
Okay, thanks, Christophe. I do have a couple questions. It's not my field, so excuse me if they're a little naive. You talked about stem cell hyperplasia being in DMD, but I know that muscular dystrophy exists in many forms. So do other forms of muscular dystrophy also result in stem cell hyperplasia as well?
That's a good question, and I don't know the answer to that question. It's not something that we have explored, but you're right that there are other forms of muscular dystrophy, and they're all related to mutations within components of the dystrophin complex. But how that would impact stem cell function, I don't think the field is quite there yet to understand how all these various mutations are impacting the stem cell. I guess the role of dystrophin in satellite cells is still relatively new, but that's something we should look at. Yeah.
And then the second question that I had is you did look at P38 gamma depletion, and you were able to show some beneficial outcomes. I was wondering if there's perhaps a pharmaceutical approach. I know that you mentioned that that's probably one of the next steps, but could there be a pharmaceutical approach to maybe interact with the pathway that you just described to have a similar outcome?
Yeah, that's a great question. And I guess I should have highlighted when I presented that experiment that's a very, that's an in vitro experiment where it's a very transient depletion of P38 gamma. And we did that specifically to show proof of concept. But when we actually tried to do this in mice and genetically delete P38 gamma, we did not see a rescue. And I think that's probably due to the multiple roles that the kinase plays in the cell. So you're right, we want to see if we can design a therapeutic approach. And I think what we want to do ideally is to promote CARM1 nuclear translocation. And so we're working on things like trying to identify the phosphatase that takes off the phosphorylation on CARM1, for example, because that would then promote its nuclear translocation. So those are the types of approaches we are aiming for, rather than like a kinase inhibition.
Okay, great. Thank you. All right, any other questions for Natasha? Okay, so if not, thank you again, Natasha. Very interesting talk. Very great work.
Thank you.
All right, so that concludes our session today. So let's again go to our screen. So that concludes our session today. So I was very impressed with all the talks today. And again, I'd like to congratulate all of the awardees for the 2020-2021 year. We are continuing to do this Young Investigator Award Symposium and also special topics issue. So please make sure to spread the word. And also for all the participants today, we hope that you continue to think about cells, tissues, organs as a venue for your research in terms of submitting manuscripts or also possibly leading future special topics issues. So on behalf of the editors-in-chief, Dr. Shang, Dr. Vibon, and myself, and the great staff at Karger Publishers, again, Nadine for her help, and also Simon Turner, I would like to again congratulate all the awardees that presented today. And so we look forward to your future research success and have a great rest of your day. Thank you again.
And thank you, Ryan, for all the work. You've been the initiator of this initiative, and it's been great. Great.
Great.
Thank you.
Yep. Thank you, Christophe.